So yes, on the days of the exams, we will not be meeting in Zoom. So there won't be a Zoom meeting on those days. So you will just be taking tests. All you'll be doing is taking tests on those days. So there's no lecture or anything like that. You just have the 4.5 hours to schedule your time however you want to do it of the class time to be able to complete those 2 exams. So yes, we won't be on Zoom. And like the chemistry quiz, like the Proctorio, the syllabus quiz, you'll be doing it on Proctorio. So you will be using your camera and be recorded, and all of that will be the same as it's been for those. So that's how you'll take those 2 exams.
And it has to be between, like you said, between 8 and 12, something like class time, correct?
Yes, 12:35. Yeah, our class time is from 8 to 12:35, so they have to be completed during that time. So like I said, what that means is if you start the lab exam at noon, that lab exam closes at 12:35. So you would only have 35 minutes to be able to complete it.
Okay, and you cannot start any earlier, like at 7:30 or something like that, correct?
Correct. It has to be during the class time. That is
Okay.
correct. So it started at 8. So Madison, to answer your question, the amount of time varies on the length of the exam, but for this first lab exam, it's probably going to be somewhere between 60 and 75 questions. So you'll probably have somewhere between 70 and 90 minutes to be able to complete that. The lecture exam is a combination of multiple choice questions, fill in the blank questions, and essay questions. So so it'll be somewhere probably between 2 hours and 2.5 hours that you'll have to be able to complete that. So if you think of the 2 extremes, 90 minutes for the lab exam and 2.5 hours for the lecture exam, that's 4 hours of test time that you'll have. And our class is 4.5 hours. And as I was mentioning, just because you have 2.5 hours to complete the lecture exam doesn't mean you have to use all of that 2.5 hours. If you are able to answer them in an hour and a half, and again, 2.5 is an excessively long period of time. No one should be butting up against the time of that. In fact, most people should be able to finish all the lecture exams in less than 2 hours. But again, because of the technical issues, because you have to scan the room, because again, I don't want the process to get in the way, I've given you guys a little bit of extra time to help with that. All right, so that is that. So there were a couple other questions that I wanted to answer and address. The first, this new one just came up. The recorded lectures, there is a YouTube channel. So again, in your modules, under the study tools, there is a link to the class YouTube site. And all of the lectures are posted on there, usually the day or the day after we have them in class, depending on how long it takes and what else I've got going on. All right, the second question I want to address as we're going through the game plan for today and the schedule upcoming. So again, today we are going to move up to the second level of our hierarchy and talk about cells. We will in lecture talk about cell anatomy a little bit, and then we'll talk in lab about our first major physiological process, and that is going to be mitosis. Tomorrow, you have your Labster on mitosis due, and that's actually the thing that I wanted to address is the Labster assignment. As was mentioned, like the unit reviews, I'm grading the Labsters for completeness. I'm looking for effort and time. And the way you show me effort and time on the Labster is that you complete 100% of the activity and you do it with at least 80% accuracy. Now, Labster is a new program that has been added to and purchased through the school for Canvas. And so what is nice about it is that it seamlessly syncs with Canvas so that when you complete those activities, you don't have a printout or anything like that that you have to worry about. It automatically submits it. However, as I've noticed, it does prorate the scores. So if it was I have set the assignment for 20 points. And if you got 90% on Labster, it gives you 90% of those points. So you would have gotten 18 instead of 20. And in fact, it does fractions. So you may have gotten like 18.4 if you got 92% or something like that. What I will say is, again, those scores are input automatically by Labster. I will be going back and manually correcting those. So as long as you completed 100% of the activity with at least 80% accuracy, you will get the full 20 points. So I know it may not show that way in your scores right now. I didn't want to mess with the scores until the assignment was due. Now that it's due, I will go back and change those. So you will get the full credit for doing that. And the same thing will happen for the mitosis one that is due tomorrow and the membrane transport one that is due Monday. So again, as long as you complete them at 100% and get them at 80%, then you will get full credit. So don't worry about what the scores say now, as long as you have that. And again, for those of you who ran into this challenge a couple times, remember your dashboard shows you your results of what you've done. To actually get to the activities, to do the activities, you actually go into the modules. And in the modules is where there's the link directly to the assignment. So you get to the assignment through the modules. The dashboard just shows you the activities that you've already completed. All right. So that's due on Friday. The mitosis is due on Friday. The membrane transport is due on Monday. You also have the Unit 3 review due tomorrow as well. Again, remember that's the wonky one where the review questions are mixed in with the, well, they're not really mixed in, they're at the top of the pages of the entire review thing. For full credit, I am looking that you have completed the review questions. I think there's a total of 10 at the top of the pages from page 62 to 68. So that is what you're doing. Fill those out, complete those, and that will get you full credit on your Unit 3 review. However, as I mentioned, even though we don't have microscopes, you are responsible for understanding the parts of a microscope, understanding the magnifications, understanding how the microscope works and changes the image. So while I want you to read through the activities, and obviously you're not going to be able to do a cheek scrape or anything like that, I do want you to read through the activities and understand the activities, because understanding the microscope is part of the exam. So you are responsible for that information on the exam. So make sure you are familiar with it at least, but you don't have to go through and fill out every activity, because obviously some of those it's not possible for you to do. All right, then you get a nice long 2-day weekend. We come back and you have your third Labster assignment due, membrane transport. Also, you have your Unit 4 lab review that is due. And again, now we're back to the normal ones where the reviews are at the end and they have the 2 parts, the check your recall and check your understanding. So make sure you've done on that. The other thing we will be doing on Monday is I have a practice lab exam. This practice lab exam is not going to use Proctorio. It is not graded. It is 0 points. This is merely 2 things. This is an opportunity. Again, one of the things that I've said to you is that I understand that this class is hard, and I love that this class is hard because I don't have to be tricky. So what this practice lab exam is actually 20 questions. All questions are questions that have been on previous exams. Some of them could even show up on your exam. And the point of this is for twofold. One, so that you can see what a timed lab exam is like on this, because again, most of you have answered multiple choice questions before, most of you have answered essay questions before, but you may not have seen a lab exam like this before. So this is going to give you an opportunity to see what the questions will be like, and it's also an opportunity, because Monday is going to be 1 week from our exam, for you to be able to gauge how well you're studying this material. If you do well on this practice exam, then that means that you're gaining the information in the right way. And if you don't do well on it, then that's going to be an opportunity for you to then adjust how you are attacking this material with a week time left to still be able to be successful when you do it for real. So you will take that practice exam on Monday, and then we will talk about it on Monday as well. I think what we'll do is we'll lecture first, and then after the lecture, I will release the practice exam. You guys will have the opportunity to take it. It'll take like 20 minutes or something like that you'll have to take it. And then we will actually talk about it after you take it, so that you can understand why I've picked those questions and what the right answers are, and to help you to, like I said, prepare for the real thing. All right, then also next week, and so again, this weekend is a good time to take an opportunity to work on this. Your PhysioX Exercise 1 is due. PhysioX is our other big lab simulator. Again, like the Labster, it does a pretty good job of walking you through it step by step. Like the Labster, it's not particularly hard, but it is a little bit time consuming. In fact, it actually baby steps you a little bit more through it than the Labster does. The only issue with the PhysioX is the PhysioX doesn't sync with Canvas the same way. So again, I know I did it really briefly on our first day on Monday when we went through it, but remember what you're going to do with that PhysioX is there are all these activities you're going to do, pre-labs, activities, you're going to collect data, post-labs, there's some review questions at the end of that, and as you go through it, you submit all of those to a lab report. At the end, you end up with a lab report, which you will then print as a PDF. And then that PDF of that you will then submit in Canvas. So in Canvas, under the PhysioX exercise, not only is there a link to the assignment, but there's a link to submit the files for that as well. Again, when you go through the PhysioX, it will let you go exercise to exercise, exercise, exercise. It will let you go through all of them. But what you need to remember is that it only keeps one lab report at a time. So if you do exercise 1 and exercise 2 and then exercise 3, and at the end of 3, you print the lab report, you're only getting the lab report for exercise 3. So make sure after each exercise, you print the lab report. You will end up with 5 lab reports, and all 5 of those lab reports need to be submitted to Canvas. So you need to submit those files to Canvas as PDFs. So again, so you will go through that. Again, it's a little easier than the Labster activities. The questions aren't quite as challenging, I found, and but it's still worthwhile activity to do it. It just doesn't, for that one, it doesn't seamlessly sync. So you will need to submit those lab reports. So make sure you do that.
Is that also completion gets full credit, or that's different?
Yes, no, exactly. All of these things, because again, most of these are learning activities. There will be some special reviews. There will be some special assignments that I will give you that will be graded for correctness.
Now you will let us know about those, right?
Yes, I will always let you know about those ahead of time. Yes, and we haven't hit any of those yet.
What if you don't have access to a printer for the PhysioX assignment?
But you say, so when here, let's actually do that. Hold on.
So I think what the professor is saying, when you click print, you don't actually print it. You click print, and then it gives you the option to save it as a PDF file. So you save it to, you save it to your computer, and then when you're trying to submit it, you submit it as a file from your computer.
Okay,
right, and we went through it really fast. So I do. So yeah. So exactly. When you go to when you go to the PhysioX, you will say when you click print, but then when you print, you select a PDF printer, or select PDF to print it as a PDF, and then what you're going to do here under the modules. So again, here are all the lab simulators. These are the Labster things, and again, but here, notice with the PhysioX, not only does this is where you are going to submit your assignment. So again, remember we've already submitted this. I did that when we practiced, but this is when you will add the rest of these to it as well. All right.
Yeah, I think so.
Yep. Perfect. Yeah. No. So you don't actually have to, kill a tree to print these things out. So that is not necessary. All right. And now, if you wanted to do that and then take a picture of them or scan them and do that, that would be fine too. So whatever makes you happy. I need PDFs submitted, and how you do this is completely up to you. All right. Any other questions on any of that stuff? All right, excellent. With that, then, we have our game plan. Let's go ahead and dive into the material. And as I mentioned, our goal now is going to be to work on our second level of organization, and that is our cells. I need my annotation code. There we go. Perfect. Excellent. Now, as we talked about when we were talking about our hierarchy, one of the big keys with cells is the first level where we actually see and talk about life. All living things are made up of cells, whether you're made up of a single cell or, like ourselves, made up of trillions of cells. One of the big differences between us and a single-celled organism is if I have a single-celled organism, oops, I need a color, there we go. If I have a single-celled organism and basically I take a big knife and I cut that single-cell organism in half, what do I end up with?
2 sister cells or something, or no?
Yeah, basically those 2 halves of the cells will, pinch off and then go and live happy lives. So I cut that off and 1 cell becomes 2 cells. Does the same thing work with you? If I cut off my right hand, does my right hand run off and live a happy life as a result of that?
No.
No, and the reason for that is that that single-celled organism is specialized to, I mean, is capable of doing all of the characteristics that we associate with life. Like we talked about, being able to metabolize, being able to grow, being able to divide, being able to respond to the environment. All the things that we talked about that we associate with life, that single cell is capable of doing. The cells in our body have become specialized. They become reliant on each other. The single cell now is not capable of doing all the things that are necessary to maintain life, right? It allows them to become specialized and work together, but it also means they become dependent on each other. So that's why I can't cut off my hand and go live a happy life, because without the help of the other cells, those cells are now, independent and dependent, are no longer able to do the things they need to do. All right. And when we talk about specialized cells, humans have over 200 different specialized cells in the body, right? You can start counting them out. We got eyeball cells and toenail cells and liver cells and so on and so forth, over 200 different types. And then when you think about plants, plants are a completely different type of animal than animals are, right? They have completely different types of cells. Other animals have different types of cells. There are hundreds and hundreds of different types of cells that can be found and identified, right? Here's the pretty picture from your textbook that talks about some of the different types of specialized cells. We have sperm cells, the only human cells that are capable of propelling themselves through space. We have nerve cells. Some of these nerve cells can be over a meter long, right? If you think about it, there are some of these nerve cells that start in the small of your back and go all the way down to your big toe, right? If you think about someone like LeBron James, that's like 47 feet long, right? Smooth muscle cells, epithelial cells, red blood cells. We have all these different specialized cells. And like I said, other animals have different types of cells, plants have different types of cells. So there's a tremendous amount of specialization. We're going to talk about a basic cell, but that basic cell can be specialized many different ways to do many different things. Now, of all these hundreds and hundreds of different types of cells that make up plants and animals and us, essentially, they can be distilled down into one of two main types of cells. What are the two main types of cells that you have in your body?
Red blood and white blood?
Well, but see, those are definitely types of cells, but again, those are part of a single type of cell. If you took all the trillions of cells in your body, you could divide them into two categories. They would either fall into one of two categories, one of the one or two categories that all the trillions of cells in your body. There you go.
Prokaryotic?
Well, remember, in humans, all of our cells are eukaryotic, but as I see it here on text, some of them are somatic.
Oh,
Somatic cells are definitely some of them. So let's define that. What do we mean by a somatic cell? What does soma actually mean? Anyone know what soma means? Really? Soma means body. So basically, somatic cell's job is to make up your body. Well, isn't that what all the cells in your body do? I know Ariel spoiled it for us, but no. There are a different type of cell, and that different type of cell in the body is a sex cell. And what is it that sex cell does?
reproduces?
True, or again, if we want to be consistent with the definition, you're absolutely right. When we reproduce, what do? We make someone else's body. So if you think about it, all the cells in you have one of two purposes. They're either a somatic cell whose job is to make up your body and make your body functional, or their job is to make somebody else's body and make their body functional. So all of our cells in our body fall under one of two categories: somatic cells and sex cells. All right? Questions on that? All right, now again, as we talked about, what's special about our cells are the smallest units that are capable of performing all of the vital physiological functions. Again, just to rattle off a few, they can respond to their environment, they can grow, they can divide, they can metabolize, and so on and so forth. These are the things that are alive. However, one of the important things to remember is that each cell is responsible for maintaining its own homeostasis, keeping itself alive and healthy. But the other thing we need to recall is that all cells come from the division of pre-existing cells. right? You came from a single cell. We have a special word for that single cell. What do we call the single cell that you came from? Right? Mom took her puzzle piece, dad took their puzzle piece. You had the spermatozoa, you had the ovum. They put them together, and when they came together, they became a very special cell. What is that special cell called? There you go. That's the magic word, a zygote. Excellent. They became a zygote. And that one zygote then divided to become 2 cells, 2 became 4, and so on and so forth to become the trillion-celled organism that you are right now. Now, obviously, all right, unless you're pregnant, nobody here has a zygote inside of them anymore, but we still need to be able to make new cells if you injure yourself or just when you're growing and doing things along those lines. We have to come from pre-existing cells. And those pre-existing cells then that help to form the new cells of your body, we have a special name for. What do we call them? What are the cells that make new cells for your body called?
Gamete?
True. Again, gametes make other people, right? Absolutely. Your
cells?
gametes make other people. But if you cut yourself, you're able to heal. If you break a bone, you're able to heal. Why are you able? You were able to grow from the size you were to a baby to the size you are now. And you were able to do those things because you were able to make new cells. And the way you were able to make new cells is with the magic term stem cell. Now, I know, again, some of the hesitation from this is when people say stem cells, other people always want to grab pitchforks and want to grab, torches and are all upset about the idea of stem cell. But that's one of the reasons why I want to take some time to talk about this. I want to be more clear about our definition. A stem cell, basically, there are 2 keys to the definition of a stem cell. The first key definition to the stem cell is that it is an undifferentiated cell. What the heck does that mean? What does it mean to be undifferentiated? Not undifferentiated. What does it mean to be perfect. Part of it is that it's not a specific type, absolutely, and it doesn't have a specific job, exactly. Stem cells are kind of like millennials. They just sit around doing nothing. They don't really have a specific type job, right? But what they are capable of doing is dividing rapidly. Oops, if I spell it right. To make new cells. That's it. That is how we define a stem cell. A stem cell is an undifferentiated cell that is capable of dividing rapidly to make new cells. All right?
Quick
Now,
question.
yes.
Now you're talking about stem cells. Are stem cells, do they decide if they're going to become sex cells or somatic cells?
Yes, partially, because, and that's the second part of what we need to talk about here, there are different types of stem cells. Different types of stem cells are going to have different jobs. For instance, the stem cells that make new gametes, make spermatozoa in males, make the eggs, the ova in females, are cells that divide to produce new cells. Kind of like your skin. Your skin has stem cells in it as well, so that when you scratch yourself or you injure yourself, you're able to produce new cells. And that stem cell in your skin, it is undifferentiated. It divides rapidly to produce new cells. But when that occurs, how many different new things can it become?
Can you repeat the question?
The stem cell in my skin. There's a stem cell in my skin here that is able to divide to produce new skin cells, which again, if you think about it, makes a lot of sense, right? Because what do we know about our skin? What do you know about the top layers of your skin?
They're dead cells?
They're dead cells, absolutely, and they're constantly being sloughed off, right? While you're sitting here, look at the dust on your computer screen. Look at the dust on the countertop next to you. The majority of that are dead skin cells from you and the other people who are around in the house with you. All right. So we are constantly losing millions of new skin cells, millions of old skin cells every day. So we have to make millions of new skin cells. Luckily, we have a stem cell here in our skin that is able to divide, and these cells make millions of new skin cells every day. And this stem cell is an undifferentiated cell that divides rapidly to make new cells. And the new cells that it makes, how many different things can that cell become?
Just one.
Just
right?
one, exactly. What we call those are unipotent stem cells. Unipotent stem cells are stem cells that can only become one type of cell. All right. In our bone marrow, inside of our bone, we have some stem cells in there that are capable of producing all of our blood cells. That means when that cell divides, the new cell that is made could become a red blood cell. It become a white blood cell, of which there are 5 different main types. It can even become the cell that makes platelets. Notice that means there are multiple things that it can become. But can that cell in your bone marrow become an eyeball cell or a liver cell or even a skin cell?
No.
No. So notice it has a limited range of things that it can become. And we call that a pluripotent stem cell. Oops, if I spell it right.
Now, or are all the stem cells, they have their own identity based on the location in the body?
Both. They have their own identity and typically they have their own location in the body. All right. So every single one of us are able to divide, are able to repair, are able to mature, are able to grow, because we all have a ton of these unipotent and pluripotent stem cells in our body. All right. So every single one of us have stem cells inside of us. The reason people tend to like to grab their pitchforks and their torches when we talk about stem cells is because there is a third type of stem cell. That third type of stem cell is an omnipotent stem cell. And of course, omnipotent stem cells, what can they become?
Anything?
Omnipotent stem cells are capable of becoming any type of cell in the body. And typically, omnipotent stem cells are just found in embryos and fetuses, right? It's one of the reasons why if you have a baby now, one of the things that they will do is you can actually do one of 2 things. Your umbilical cord blood has some of these omnipotent stem cells in it. So some people will donate that to science so that scientists can use these omnipotent stem cells to do research. Other people will actually freeze it and store it, thinking that maybe by the time that, you know, that your baby becomes a mature adult at 30 and 40, right, maybe they'll become a horrible drunk at that period of time and have ruined their liver with liquor and alcohol. However, technology will advance to such a point where we'll be able to use those stem cells of them to grow them a new liver and plop it in their body. Right, and so again, so those cells have the potential to become anything. They're very valuable for research. The potential for them is truly unlimited if we can learn how to harness them. But what the problem with those omnipotent stem cells, as I said, is they're typically associated, well, they're only found in embryos and fetuses, and then so obviously you need to get them from embryos and fetuses, and then there's the whole social connotation that goes along with that. So that's why stem cells tend to be such a highly potentially, volatile subject for people to talk about. But the important thing to remember is you are able to grow, you are able to mature, you are able to heal, because everybody has stem cells in the body. There are different types. Yes, embryo and fetuses are the only, as we are developing as embryos and fetuses, are the only place where we have omnipotent stem cells. Basically, by the time we are born, all of our cells have become differentiated enough where they're just going to be unipotent and pluripotent at that time. Yep. All righty.
Now, can those cells, can they be given to anybody? Like if I have it, can I give it to you, or does it only work for myself?
Well, so again, the same way that you can't just get a liver from everybody. There have to be compatibility issues with that and things along those lines. But again, we are not in the stage where medically we are capable of doing these things. However, in a strictly theoretical sense, right, omnipotent stem cells potentially would be something that could be given to somebody to help someone. So the classic example of this you see is in spinal cord injuries, right? One of the classic examples, and I know I'm dating myself a little bit this, and most of you don't remember this, but back in ancient times, there was an individual by the name of Christopher Reeves, who was an actor who played Superman in several movies, very famous, very popular guy, a very strong PETA proponent. And then one day he was out on his horse and got bucked off his horse and had a spinal cord injury and ended up in a wheelchair, and suddenly became a huge proponent of stem cell research because some of the potential goals, as we know, nervous tissue is one of those tissues that doesn't regenerate. So theoretically, if you could take some of these omnipotent stem cells and input them into the body, they could then grow to produce the new neurons to replace the neurons that have been injured that your own body isn't capable of being able to produce. Now, obviously, we're not anywhere near there, and Christopher Reeves ended up dying in a wheelchair, never being able to walk again. But again, theoretically, the potential is there, and so there's a lot of research and time going into it. But because these only come from embryos and fetuses, obviously there's not the debate as to when life begins is also a very deep and important philosophical discussion. But also there are concerns that, especially with poorer and underrepresented populations, that as the demand for these types of cells become more prevalent, there could be medical pressures to put on people to terminate pregnancies that they wouldn't normally terminate just so they'd be able to get the cells. So again, there's huge social, racial, religious connotations that go along with these. And so that's why, like I said, stem cells tends to be a very charged topic. But the important thing to know is, as we are talking here strictly from a biological standpoint, right, we have a biological definition of what a stem cell is, and appreciate that everybody has stem cells in their body. Everybody doesn't have omnipotent stem cells in their body, but we know what a stem cell is, and we understand how stem cells work. All right?
Excuse me, Professor, I have a question.
Yes.
What cells did you say that cannot regenerate again?
Nervous tissue.
Okay, thank
Well,
you.
we have several. Nervous tissue is one of them. So again, and primarily I mean like brain and spinal cord. One of the things that happens, for instance, this is normally when we're in the classroom, this is easier to show off. I have 2 large scars on my knee because I had to have major reconstructive surgery on my knee. And after the surgery, my first surgery, there was about a silver dollar size space on the side of my knee that was completely devoid of any feeling because the nerve had been cut on the way there. Now, as long as the cell body of that nerve and the neuroglials and things like that are intact, and it took several years, but I was actually able to regrow the nerve to that region, and I now do have feeling on the side of my knee there. However, with the brain and spinal cord, whether it's a traumatic brain injury, whether it's falling off your horse or things along those lines, typically when the brain and spinal cord are injured, that nervous tissue is not repairable. It doesn't regenerate. Right? In fact, and again, we'll talk about this today when we talk about mitosis. What's the other major tissue type in your body that does not regenerate? Usually someone knows it.
You mentioned brain cells, right?
Yeah, so nervous tissue is one. What's the other big tissue type in the body that does not regenerate? Think of it this way. There you go, bingo. Heart, cardiovascular muscle. The muscle of the heart, when it becomes damaged from lack of oxygen, right, you end up usually needing, if it's severe enough, a pacemaker because it gets disrupted, it disrupts the normal cycle, and that tissue doesn't heal. So yeah, typically cardiovascular muscle or cardiac muscle and nervous tissue are ones that don't regenerate.
So, question. I
Yes.
have an understanding if you had a heart attack, your heart would actually grow arteries or veins around it so that it would keep oxygenating that area. Is that not this?
Yes and no. So when there is, depending, so part of it is depending on the severity of it, yes. But if there is a blockage to a blood vessel in the heart, your heart does have the potential to be, and all parts of your body have the potential to grow new blood vessels to circumvent that blocked area and get to the tissue and bring it the oxygen. But think of the time that it takes to grow those blood vessels, right? And the time that it takes to grow those blood vessels, all that muscle is going to be deprived of oxygen in that case, and it's going to become permanently damaged and it doesn't regenerate. So when someone has a blockage, instead of waiting for the body to regrow those blood vessels, typically what we do is a heart bypass surgery, right? Where they actually go in, they harvest blood vessels from either a cadaver or often from parts of the leg, and they will basically put in a shunt basically to run blood, an alternate collateral pathway to run the blood to that region instead.
Okay, so it's not helping the tissue. The tissue is already dead and there's no curing that. It's just working around it so other tissue doesn't die.
Yes. Yep. All right. Right, and that's ironic. Well, as Ryan pointed out, all right, nervous tissue, cardiovascular tissue, the cardiovascular muscle are two of the most important tissues, and they don't regenerate. And that's probably one of the reasons why they are so metabolically active. Because as we're going to see when we talk about mitosis, dividing requires a tremendous amount of resources, a tremendous amount of energy. And if these cells, like your heart cells, are super active all the time, they probably don't have the time and the energy and the resources to be able to divide. All right, excellent. Great questions, great stuff. So that is our stem cells, perfect. All right, so like I said, we have the ability to make new cells. There's also a tremendous amount of variability in our cells as well. However, for the most part, we're going to talk about a typical cell. And when we talk about typical cells, because once we understand the typical, then we can talk about the specialized. Typical cells have three basic components to them. The first of those is the nucleus, right? And of course, right, sentences, words matter. Most cells have one. And of course, what is the key word in that sentence?
Most.
Most, because what does most mean?
Not all.
Not all, exactly. There are some cells that actually have multiple nuclei. And there are actually some cells that at different stages in their life do not have a nucleus. Now, of course, if you don't have a nucleus, you're probably not going to last for very long, but there are definitely some important cells, like our skin cells, like our blood cells, that red blood cells anyway, that at points of their life do not have a nucleus. As we talked about, a nucleus is that lockbox where we keep our DNA. That DNA, remember, are those genes, and those genes are the instructions for making proteins. So we have that locked away inside of it. The second component is the cytoplasm. So let's actually cheat. Draw my nucleus here and put a big N in that so we know that is the nucleus. As we also talked about, let's change the color here. We also know that, as we talked about in the last class, a cell is going to have a plasma membrane. And what is the primary macromolecule that comprises that plasma membrane again?
A bilayer.
A bilayer of what?
Lipids.
Lipids, and not just any lipid, but phospholipids. Excellent. Right, so those phospholipids that are arranged in a bilayer, as you guys mentioned. Perfect, excellent. So we have that component. And then the third component is the cytoplasm. Cytoplasm is basically everything else in between. So all the hunk and junk that is here inside of the plasma membrane, between the nucleus and the plasma membrane, is our cytoplasm. And that cytoplasm actually has two parts of it as well. There is the water part of it, and that watery part of it is the cytosol. And we'll talk about this again in a second. And then we also have our organelles. Right? The same way your body has organs, that help them to do the job that it needs to do. Our cells have organelles that help the cell to do the jobs that it needs to do. So again, I've done an amazing job of drawing one of these things here, but let's actually look at the pretty picture from your textbook. And here we can see a much better example where, again, we have our plasma membrane on the outside, we have our nucleus on the inside, and then we have all the hunk and junk in between, that is the cytoplasm. Now, let's talk about the nucleus first. Great question. So cytosol is more than just water. It is the watery part of it, but it is the water and the stuff that is dissolved in water. So it isn't just pure H2O, it is the water and all of the water-soluble things that have been dissolved inside of it. So, like I said, most cells have one of the nucleus, but some can have more. When we talk about the anatomy of this, as we can see here, we have the nucleus. The nucleus is an example of a membrane, bound organelle. Again, remember, an organelle is a collection of proteins and chemicals that are held together in space so that it makes it more efficient to do its job. And so it has a membrane, and that membrane is going to be comprised of phospholipids. And again, like all membranes, they are in a bilayer. However, one of the things that is special about the nucleus, if you look closely, it has, and let me change to here, not one, but two layers. So actually our nucleus actually has a double membrane, which if you think about it means 4 layers of phospholipids. So again, if we were to, I'll just quickly use the drawing app to do this. All right, one head, one tail, one tail, one head, right? That's one phospholipid bilayer. And then we have a second one with the head and the tail and the head and the tail and a second. So it has 1, 2, 3, 4 layers of phospholipids arranged into 1, 2 layers for a double layered membrane. It is really, really, really important to keep the stuff that is inside our nucleus, the DNA, protected. However, and so we have this double layer membrane that we call the nuclear envelope. Now, while it is very important to protect the DNA inside of here, it is also important that eventually that information gets out to the cytoplasm so that we can make the proteins. So if you notice, there are all these orange proteins embedded in a circle forming small holes in the nuclear envelope, which allow things to come in and out. And those small openings, those spaces, are the nuclear pores. So we have the nuclear envelope, which is a structure, and our nuclear pores, which are spaces or holes that are in them. Notice I'm not just picking these words or these terms randomly. These are, again, the terms that we would be using on the exam. This is one of the challenges for this exam. As I said, you got to be careful, and I'm going to be very precise. So if I have an arrow pointing like this, and again, granted, this would be a crappy arrow to do for this on the exam, but if I were to do this where it's kind of in between the two, and I asked you to identify the structure that I am pointing at, what do you think the structure that I'm pointing at is right now?
Nuclear envelope.
Nuclear envelope. If I were to ask you for the space that I'm pointing at, what would the space be?
Nuclear pores.
Nuclear pore. Excellent. Now, if I wanted to do the nuclear envelope instead, it'd make much more sense for me to put the arrow there. If I wanted to do the nuclear pore, then yeah, it would be much more sense for me to put the arrow like that. So again, I'm not going to be tricky. I will try to be obvious with my examples. So having it in between like that wouldn't be the way it would be on the exam. The point I'm trying to make is that I will use specific words to lead you to directly the piece of information that I am looking for. And so this is examples of that. The nuclear envelope is a structure, while the nuclear pores are spaces. All right. Now, as we mentioned, it contains the DNA. But what we didn't talk about is DNA doesn't like to hang out by itself. DNA by itself is brittle. So what happens is DNA has a lot of proteins. And those proteins help and assist it to be stable and to be maintained and to have a little bit of shape and structure to it. So this DNA and protein are intertwined with each other in these big, huge, loose strands. And because, as we've mentioned, anatomists love to name everything, the fancy name they've given for these big, loose strands of DNA and protein is chromatin. Now, eventually, this cell is going to need to divide from one cell into two cells. And when it does that, both cells need DNA. Now, these loose strands are like a bowl of spaghetti. Anybody here shared a bowl of spaghetti with anybody else before?
No.
Right. Well, you may have felt, you know, if you've got a loved one or someone like that, they may want a bite or something like that. But if you had a bowl of spaghetti, and especially like if you had two kids and you wanted to separate it evenly for both of them, how easy is it to divide a bowl of spaghetti into two? Well, it's pretty easy if you use a knife. You cut it in half and scoop it out onto two bowls. But you damage the strands that way. So now what you have to do instead is you have a bowl of spaghetti where you have to separate into equal number of strands in two bowls, and you can't break any of the strands of spaghetti. Is that going to be easy to do?
No.
No. Luckily, the other thing those proteins help the DNA to do is to fold up into a tight bundle. When it becomes time for the cell to divide, these loose bowl of spaghetti strands fold up into a tightly packed structure. And so what happens is when that DNA and those proteins fold up into a tightly packed structure, notice it's the exact same material, but that new material now has a different shape to it, and we give it a different name. We call it a chromosome. And as you may know, how many chromosomes do you actually have in every single one of your cells?
46.
46, there you go. 46, or the other number you might have been thinking is 23, because it's 23 pairs. You got 23 from dad and 23 from mom, right? So really, the only difference between chromatin and chromosomes is the shape. When it is loose and unfolded, it is chromatin. When it is tightly packed to allow the cell to divide, and that is the only time this occurs. You only get chromosomes when the cell is going to divide. But when it is getting ready to divide, that chromatin condenses down into chromosomes. All right? Fun with vocabulary. Speaking of fun with vocabulary, notice there's another structure in here as well. This blue structure, known as a nucleolus. Nucleolus is the singular. Nucleoli is the plural. When the DNA is a loose strand, and I'll cheat and draw a little loose strand of chromatin right here. As I mentioned, this loose strand has the information, the gene on it for making a protein. And what happens is that information needs to be read. We'll talk about protein synthesis. I think we're doing that, no, we're doing it Monday. We'll get to it Monday. We'll talk about this process, but protein synthesis, making these proteins, copying this information requires a tremendous amount of stuff. We need more nucleotides, we need enzymes, we need other special proteins. And so what happens is all these special proteins, all of these enzymes, all of this stuff wraps around the part of the DNA that is being read. When we look at this under the microscope, DNA and the chromatin, the DNA and proteins are so small, you can't see individual strands. It's going to look like a loose cloud, a fuzzy cloud of material. But these materials, where we're reading that DNA, become so dense, you will actually see it as a dark spot on a light microscope. And that dark spot means that right here in this area, we are reading the material and making proteins. And this structure that can be seen with our light microscope, where we're reading the DNA to make protein, has a special name. We call it a nucleolus. And again, cells are not required to just make one protein at a time. So when you look in the nucleus, you may see 2, 3, even 4 nucleoli, meaning that 4 different proteins are all being made at the same time. All right, so inside your nucleus, you may have 1 or more nucleoli, right? And your chromatin becomes chromosomes, right? Fun with vocabulary. Trust me, it's gonna get worse. All right, here, yeah, Ryan, you had a question? Are the nuclear pores like a hole or a gate where it opens and closes? Great question, sorry. No problem, I apologize, you're right, I missed that before. So it is a hole. It does not open and close, so it is constantly open. So it is a hole, it is a pore, right? It is constantly open. Great question. Sorry I missed that the first time. Thank you for making me aware. All right, here again is our DNA. Remember here, the outside is that sugar and that phosphate making the backbone, and it is a double helix. And again, we'll talk more about that structure in just a bit. But like I said, DNA is not by itself. There are dozens of proteins. One of the most common proteins that helps the DNA are histones. So notice the DNA is wrapping around those histones. It helps to keep it from getting kinked or damaged. It allows it to be more easily accessible. And it's also one of the proteins that helps this big loose strand of DNA pack up into this tightly packed chromosome. All right, so it helps to protect the DNA and allows that DNA to fold up. Again, as I mentioned, we only form chromosomes when the cell is getting ready to divide. And again, we have a total number of 46 chromosomes, 23 you got from mom and 23 you got from dad. All right, like I said, we'll talk about this structure of the DNA and actually with that nucleolus and what's going on there when we get to protein synthesis next week. Let's talk a little bit about the cytoplasm. Cytoplasm, as we mentioned, is composed of 3 components. The first is our cytosol, and I know I use the term water, but it's really more watery. The interchangeable term of cytosol is intracellular fluid. It makes up about 55% of the cell's volume, and it is mostly water, 75 to 95%, but it's also the substances that are dissolved in water. Things like proteins, things like carbohydrates or amino acids or ATP, or things along those lines. The other big thing that we have here inside our cytosol are, and let's actually cheat and do this first, are ions. And one of the important things to remember about ions are unequally distributed. Inside and outside of the cell. When we have a cell, and for this, I'm gonna actually cheat and go back to our whiteboard because it gives me more space to draw. Here is our cell, and we have an unequal distribution of ions. Anyone know what the most common cation ion in the body is? What do I mean first? What do I mean by cation? What is a cation?
Positive.
Yeah, there you go. Cations are positively charged. What's the most common cation found in the body?
Potassium.
Potassium's close, and potassium is the one that is the highest level inside the cell. So inside the cell, we have very high level of potassium.
Sodium?
Right, but the actual most common cation in the body is sodium. So while we have a high level of potassium inside the cell and low potassium outside the cell, it's the opposite for sodium. Sodium, there's a very high level of sodium outside the cell and very low level of sodium inside the cell. What's another important ion that we find associated with the cells?
Oxygen.
True, but oxygen is not an ion, remember. Oxygen isn't charged. You're right, oxygen is vitally important. But right now we're just talking about ions. You're right, oxygen is vitally important.
Hydrogen?
Hydrogen is a good one, although hydrogen, remember, plays a role. If you think about it, hydrogen is acids are hydrogen donators. So when we talk about hydrogen ions and hydroxy ions, those are things that are associated with acids and bases. And remember, we've talked about how important it is to maintain the pH of the blood. So for that, we do want to limit the amounts of those that are around. So we do want to limit those because we want to maintain an appropriate pH inside of the body. I thought I heard someone say it. Hold on a second. Another vitally important one is calcium. Calcium is a super important ion in that calcium makes cells do wonky things. If you are a muscle cell that you want to contract, you need calcium. If you're a neuron where you want to send a signal to another cell, you need calcium. If you're a gland that needs to release sweat because you've been running around the room, you need calcium. Calcium makes cells do wonky things. Now, if calcium makes cells do wonky things, do you think you want a lot of it inside the cell all the time? No. So there is high levels of calcium outside of the cell, and in fact, very low levels of calcium inside the cell. And of course, we've all been talking just about cations right now, but what about anions? What's the most important anion? What's the most important negatively charged ion?
Oxygen? No.
I know you know. Carbon, remember, is not an ion, not charged.
Is it, no, nitrogen and cat.
Yep. You guys are overthinking it. What are we? We're big bags of salt water, right? Isn't that essentially what we are? There you go. Chloride ions, excellent. Right? We're big bags of salt water. Sodium chloride, salt, absolutely excellent. And not surprisingly, there are high levels of chloride outside because we're big bags of salt water, and low levels of chloride inside the cell. So one of the things we have is we have this unequal distribution of ions. Some are higher inside, some are higher outside, and that inequality is what's going to allow the cell to do work. These ions and their unequal distribution, and again, that's the magic term we want to remember. It's an unequal distribution of these ions is what is going to be vitally important for these cells to be able to do work. All right, back to the slideshow. So, like I said, we have, for instance, high levels of potassium inside the cell, low levels of sodium because we have that unequal distribution of ions. Now, these are things that are true for most cells. There may be minor variations, but these are the things that are true for most cells. Other cells, many cells, are going to have things that we call inclusions. Inclusions are specialized chemicals that are going to vary by the cell. We already talked about a couple examples of these. For instance, when we talked about glycogen, remember glycogen was that macromolecule, that polysaccharide that we talked about with lots and lots of sugars put together. And again, we talked about how there's lots of it in the liver, or we talked about how there's lots of it in muscle cells. Right, so those are things that, for instance, that's a molecule that is found just in that. We also talked about hemoglobin. Hemoglobin was a special protein that is not found in every cell of the body. In fact, it's pretty much just found in one cell of the body. What was the one cell in the body that we said had hemoglobin again?
Blood cells.
Yeah, red blood cells. Because what does hemoglobin do again?
Transports oxygen.
Transports oxygen, absolutely. So that is the one cell that has hemoglobin in them, right? So again, there are going to be cells that are going to have specialized molecules that are specific to their function inside of them. And so again, because anatomists love to name everything, they've come up with a term for these types of specialized chemicals, and they use the term inclusions. It'd be pointless to rattle off the list of all of them, but like I said, like glycogen, like hemoglobin, as we come across examples of them, we will talk about them. All right, and that leads us to our last component, and the last component are our organelles. Organelles come in two flavors, membranous and non-membranous. What do you think the difference between a membranous and a non-membranous organelle is?
One is flowing and the other is in a bound place.
Excellent, I like that, perfect. Membranous organelles are bound by a membrane, whereas non-membranous organelles, as the name would indicate, are not bound by a membrane. There you go. See, not all the questions have to necessarily be tricky. So absolutely. So some are bound by a membrane, some are not. There is an advantage to being bound with a membrane. And again, this is one of those places where I normally have to, now I'm gonna have to work on my analogies. Because normally I make the analogy at this point of how we're all sitting here in a classroom together. And because we're all locked in this classroom together, when I speak at a relatively normal voice, you're all able to hear me and you're able to gain the information from me. So by putting us all together, we make the process more efficient. If instead of being bound in the classroom, you guys were able to freely roam around campus, and I just stood in the middle of campus screaming at the top of my lungs all of this information, would that necessarily be as efficient of a way of getting all of the information to you guys? No, by having us bound together in one location, it makes it more efficient. Doesn't mean it can't be done, it just makes it more efficient. So as we work our way through the organelles of the cell, the organs of the cell, we will see that some are bound by a membrane and some are not. All right, questions on that? All right, I know we started a little late on lecture because you guys had a quiz, but this is a good natural stopping point. What we're gonna do now is start working our way through the organelles, both the membranous and non-membranous of the cell, talking anatomy. So this is, again, a lot of vocabulary, a lot of information. So this is a good stopping point. Let's go ahead and take our first break. We'll call it a 10-minute break, but we'll call it closer to 12 minutes. Come back at 9:25, and at 9:25 we will pick up from here. So let's restart at 9:25, and then I will start the recording. All right, so again, this is an opportunity to go to the bathroom, go to get something to eat, drink, get coffee like I'm gonna do. Like I said, check your turnip prices, whatever it is that you need to do, this is your opportunity to do that. All right, any questions before we stop?
Yes, I have a question.
Yes.
I tried to do the, what was it, the lab last night,
Yes.
and it was saying that it just kept asking me to reload the page or something like that, but I wasn't able to do it. I want to know if anybody else had that issue.
I had the same issue.
Were you guys doing it through, were you doing it through either Chrome or Firefox, or the correct?
Yes.
It worked the second time, I think, after using Chrome and clearing all the cookies and stuff, and then going into the similar thing you said that you have to go to your modules, not just the dashboard. That
Yes.
worked.
Right. So yeah, and then Helen, let's do that again real quickly, because again, I got several emails on that. Where are we here? Yeah, I think one of the things that has been confusing for some people is that there's this Labster dashboard. When you click on the Labster dashboard, this is where it's going to give you your general information about it. And notice if you haven't done any simulations yet, there is no data for that there. So instead, what you need to do is you need to go to the modules. From the modules, then down here in our section 1, which is the section we're in now, We have all the different parts, and here is where our lab simulations are. When you click on your lab simulation, and again, for instance, like homeostatic control, it has the instructions, it has here, and this is where it loads it. Now this one, I'm not on Chrome, so again, I will get the error message because I'm not on Chrome, Firefox, or Safari. These are the only browsers that are supported. I know there are some delays in loading, so sometimes you have to be patient on it. But like I said, if you refresh it, if you close it and open it back up again, if you clear your cookies, it should hopefully work. If not, I do know that Labster's tech support is pretty good. There's over on the side window over here, there's some help things, so you can try using that if you're having difficulty, and they should be able to help you with that as well.
Okay.
Okay. All righty. Questions on that? Or anything else?
So, Professor,
Yes.
can we do it today? Resume it today? The lab for the Labster?
I'm sorry?
Can we redo it today?
Yes, you can redo the Labster, and then obviously we have another Labster due tomorrow. But yes, the Labsters, while they have a due date, I don't have opening and closings for them because I want them to be learning experiences. I want them for you to be things that you can go back to. And while there are due dates for these things for the first week or so, I am going to be kind of lenient, a little bit lenient on the due dates of these things because I know there are some technical issues. But again, I want people I want most people doing these things and getting them done and staying on task, because if I set the due date for later to give time for people to work out problems, nobody does it till the day before it's due anyway, and so then we're just gonna have problems later. So this is your opportunity to figure out those kinks and get it up and running properly. But I don't want people falling behind. So yes, you still have that opportunity to complete.
Okay, thank you. Because there will be some technical issues here. Yeah.
Okay, yeah, exactly.
Thank
so
you.
Work those out, but yeah, we need to work those out fast. That's the problem with summer. We don't have a lot of time. It's very fast paced. So that's why we have to get these things up and running as soon as possible so that we can stay on top of it. Because like I said, your test is a week from Monday on the first 5 chapters. So it's a lot of material in a very short period of time.
Professor, one question. When we're gonna be taking the exam, like maybe 1 day before the exam or 2 days before the exam, are we gonna be able to just have time only to study, or do we still have to do homework and mastering and all that?
Well, again, if you look at the schedule, you will see that there is no assignment due on Monday. So the exam is Monday, and your last assignments are due on Friday. So you do have the weekend to study. Looks like I'm looking at the schedule right now. All the exams are, well, okay, the first exam is on Monday, and all the rest of the exams are on Thursday. So you will have at least Wednesday to prepare for those. But again, welcome to summer school. Their time isn't your friend in summer school, as I keep saying. It's going to be very fast paced. So that's why it's important to stay up on these tasks, stay current with the material, because there is no time to play catch up.
Okay.
All righty. All right. So again, well, now we've talked for half of the break. So let's go again. Like I said, take a 10-minute break. That'll give me a chance to go get my coffee, and let's come back now at 9:30. So since I haven't stopped the recording yet, that we can still take a real break. So again, we will now restart at, let's say, 9:30, and I will start the recording at that point. All right. Any other questions? These are all great questions. I'm happy to answer them. Any others? As I've said before, and I will continue to say, the only stupid question is the question not asked. So I appreciate the questions. Please, if you're unclear on something, ask, because especially now, right? In the classroom, it's different. When we're walking around during lab or during the breaks, it's easy to walk up to me and ask a question then. This is our opportunity to interact. So it is really important that if you're not understanding something, that you ask those questions, because you're putting time, money, and effort into this class, and it is important that you get what you need out of it. All right.
Are we going to be talking about the sodium potassium pump today?
No, we'll get into that tomorrow when we talk about the plasma membrane.
Thank you.
But it is the most common. If you have a specific question about it, I'm happy to answer it. But we will talk about it. I do promise.
I promise you I'll have questions for you tomorrow.
Excellent. All right, sounds good. All right, so I will see you guys back here at 9:30 then. So Our next goal, as I mentioned, is as we work our way through the cytoplasm to talk about the organelles. We'll actually start first with the non-membranous organelles. Yes, Ryan, do you have a quick question?
Sorry, could I ask a question pertaining to stem cells again?
Yeah, sure.
I was curious, what makes differentiation happen, or how does it become identifiable as a specific cell?
So the short version of this, let's do it two ways. Let's talk first about the pluripotent stem cells, because pluripotent stem cells, like the ones in our bone marrow in our blood, are capable of becoming any of the cells associated with our blood. So when that cell divides and produces a new cell, the question then becomes, all right, how does it know whether to become a red blood cell or a white blood cell? And with white blood cells, there's 5 different types. How does it know which type? And how does it know? And the answer to that is chemical signals. Some of those chemical signals are hormones or other hormone-like proteins. Typically, chemical signals are what going to determine what that cell can become.
And so when it becomes a specific cell, then it starts making specific proteins or something?
Yeah, absolutely. So
what
what'll happen is that cell becomes what we call a committed cell, where now its destination has been determined. So that hemocytoblast, that cell in the bone marrow, forms a new cell, and that new cell basically becomes a committed red blood cell.
Okay.
And once it becomes a committed red blood cell, one of the things it's going to start doing is making massive amounts of hemoglobin.
And then
that's going to be its job.
Stem cells have all the same internal organelles as other cells, right?
Yeah, absolutely.
Okay.
Yep.
Thank you.
Yep, no, good questions. All right, so let's talk about those organelles, starting first with our first non-membranous one called the centrosome. The centrosome, as most prominent feature, as you can see, are these 2 tubular structures here known as the centrioles. These centrioles are very distinct in that they are always perpendicular to each other, and they're formed from large proteins of tubulin proteins, so they're tubular proteins. These centrioles are sometimes also referred to as the microtubule organizing centers, because guess what it is that they do?
Organize protein packages.
Well, not just that, but they organize microtubules, right? They're microtubule organizing centers, absolutely. As we'll learn, these microtubules form things that are known as spindles. And those spindles are going to be important for moving the chromosomes around when the cell divides. What they've actually been able to do in animals, oh, I lost my annotation, there it is. In animal research, they have actually been able to take a laser and in animal cells gone in and destroyed these centrioles. And when they destroy these centrioles, what ends up happening is that cell can no longer divide. So perfect. These are the microtubule organizing centers. They're required for cellular division, end of story. And then one scientist, just on a kooky whim, said, hey, you know what? Plants have these centrioles as well. I bet if we destroy them in plant cells, they won't be able to divide as well. So they went and destroyed them in the plant cells, and guess what happened? Plant cells divided. Yeah, the plant cells divided, exactly. So for whatever reason, these are required for cell division in animals, but not in plants. Now, notice, exactly, life is odd. Notice centrioles and centrosomes, though, are not identical terms. They don't mean the same thing. Centrosome is the organelle. All right? Centrioles are structures. And there is an additional structure, or really an additional component to this centrosome. And that is, and I'm going to use my highlighter to cheat this. Notice around the centrioles is this kind of yellowish cloud-like looking material. This yellowish cloud-like looking material, as you can see here, is known as the centrosome matrix. So basically these centrioles are surrounded, and sorry, I got my pictures in my chat in the wrong place. I need to cheat and move those out of the way for a second. This centrosome matrix is, we could call it a structure, or really it's probably more appropriate to call it a substance. Because basically what it is it is a thick cloud of dense proteins that surrounds the centrioles. It is not held in a membrane, so how it stays condensed and packed together is not fully understood. But we do understand that this dense cloud of matrix surrounds the centrioles, and those things together form the chromosome. Again, this is kind of wrong. Let me finish my thought and then I'll answer your question. The important way to think of this is to think of it in terms of components and full structure, right? You have a table. That table is made up of 4 legs and a flat surface. So the whole thing together is a table. The parts of that table are the top and the legs. Well, here we have a centrosome. The centrosome is made up of 2 parts, the centrioles and the centrosome matrix. So the structures make the whole. The centrioles and the centrosome matrix makes the organelle. That is the centrosome. All right, Ryan, what's your question?
So this image of the centrioles, are they little churro looking things, or?
They are indeed little churro looking things.
Okay.
But again,
So that's not a cut.
No, it is no, they're not cut. This is
Okay.
the length of them. This is what they look like. And again, what you have to remember, and this was actually kind of the next point I was going to get to, is this is one artist's representation of it. If we were in the classroom, this would be the point where I would get out our Elmo camera, and what I would do is show you this. This is our cell model that is in the classroom.
Mm-hmm.
Notice again, in this we can see some of the key things we've been talking about already. Notice here we have our nucleus. We can see there are 2 layers to the nuclear envelope. We can see the nuclear pores that are in between them. And lo and behold, and I'll put a circle around it so we can see it clearly, we have 2 churro-looking perpendicular structures that are side by side with each other. What structures do these 2 things represent?
The centrosomes?
The centrioles?
Centrioles. Remember the key word there I said was the structures. Structures
yes.
that they represent are the centrioles. Now, this particular artist, have they bothered giving us a representation of the centrosome matrix?
No.
No. The most prominent part of the centrosome are the centrioles. So they've given us the centrioles, and because they've kind of thought of the centrioles and the centrosome as interchangeable. They're not. Luckily, this isn't the only model maker. We have a second model in the classroom, and here is our second model. Now, this picture isn't quite as clear, but notice what we can see. And Madison, I see you just, I'll answer your question in just a second. Notice here are 2 perpendicular, kind of fuzzy, but churro-shaped structures. Guess what these yellow structures are?
Centrals?
These are the centrioles. Excellent. And notice here, they've put, now again, they've put a very distinct border around it, almost making it look membrane bound. But notice here, this is membrane bound with a separate color on the outside. This doesn't have a separate color on the outside. So guess what this black substance represents?
Centrosome.
Yeah, the centrosome matrix. Excellent. So again, we have these different artistic representations of these things. So that's always going to be a challenge we're going to be dealing with. But remember also, yes, the centrosome is the organelle. The centrioles are the structures. I would say the centrosome matrix could be a structure or a substance. And often on the exam, will actually write it that way. Because again, I want to be clear. So notice I could have an arrow on the exam. And this is again where part of the challenge comes in this. As I've said before, people often lose points, not because they don't know the information, but because they don't read the question carefully. Notice I could have my arrow. This could conceivably be the very first question on your lab exam. I guarantee this picture and the previous picture will be on your exams, pictures of the models will absolutely positively be on the exam. So I guarantee this will be, well, okay, let me rephrase that. Because again, we're online, everybody gets a different random exam. I guarantee in the pool of questions that are you are responsible for on this exam, there will be pictures of this model. Will one of them randomly show up in your group? That I don't know. I guess technically it might be possible you could take your exam and your random collection might not include this picture. But I guarantee it's in the pool. And as I said, people often lose points not because they don't know the information, but because they don't read the questions carefully. And the reason for that is I can have an arrow like this, and there can be multiple, 2, 3, sometimes 6 different questions that I could possibly ask where the answer is going to be different. With this particular arrow, there's basically 2 questions I could ask. I could ask you to identify the specific organelle, or I could ask you to identify the specific structure. All right? What would the answer to question 1 be?
Centrosome?
Centrosome. What would the answer to question 2 be?
Central?
Centriole.
Centriole. Excellent. Perfect. I could also instead move the arrow here and ask those same 2 questions. Although, to be honest, for this second question, I would likely word it in the way where I'd say identify the specific structure / substance. When I do that, and again, this doesn't fit on the screen, so hold on, let me cheat. I'll do that, and then I can do, oops. There we go, that kind of looks like it works. Excellent.
Works.
When you see word / word, that doesn't mean I'm asking for 2 things. When I ask for 2 things, I will specifically say, give me 2 answers to this question. What I'm trying to do here is use all of the possible correct vocabulary to make sure that you understand what I'm doing. Right? Because like I said, it is a cloud of material. So it's kind of a structure, but it's also kind of a substance. Both of those are kind of right. So rather than use one term that might be confusing to you, I will make sure I use both. And so again, if this was the question, and these were the 2 possible, I mean, if this was what the slide looked like, and these were the 2 possible questions, what's your answer to question 1?
Centrosome.
Centrosome.
What's your answer to question 2?
Same.
Centrosome. Yeah.
No.
Centrosome.
No?
Question 2, the answer is what?
Centrosome.
Centrosome matrix. Absolutely.
Oh.
Centrosome matrix. Right? So the structure / substance is the matrix. Okay? Notice if we go back a slide, I can't ask this question. All right? Because this one doesn't have a matrix.
Mm-hmm.
So obviously, you know this slide will not be, you will never, the correct answer to the question on this particular slide is never going to be centrosome matrix because this one doesn't have one. Does that make sense?
Mm-hmm.
Like I said, my goal of this is not to be tricky. Oops, wrong button. My goal in this is to be precise so that you can give me the precise piece of information I'm looking for. And again, the challenge in this is that I could have an arrow like this, and because we just finished talking about it, this is a fair way to ask these questions. I could ask you any number of things. I could first ask you identify the organelle. What's the answer to that question for this arrow right now?
The nucleus.
Nucleus, excellent. I could ask you for the structure. What is your answer for the structure?
Nuclear envelope.
Nuclear envelope, excellent. I could ask you for the specific macromolecule that forms this structure. And what would your answer for that be?
Phospholipid.
Phospholipids. I could ask you how the macromolecules that form this structure are arranged. And your answer to that was?
Double bilayer.
Yeah. And it would be a double bilayer. Excellent, right? I could cheat and move my arrow just a teeny bit like that, and I could ask you to identify the space.
Nuclear envelope.
Space.
No, nuclear
Pore.
Nuclear pore.
Emerald nucleus.
Nuclear pore. Excellent, right? So again, I will be very precise in the wording of my questions, and I want you to be equally precise in your answers. Again, the one nice thing about this setup is normally when we're in the classroom, you get one question for 45 seconds, and then the next one comes up. On this exam, you will see all the questions at first. So you'll see a question, you'll see all the questions, you can go through them in whatever order you want, you can go back to questions if you have it, so there is a slight advantage to that. But people still make the same mistake. They get excited because they see an arrow pointing at something that they know, you know, like right there, and they go, oh, that's the nucleus, I know that. And the problem is the question is asking, what is the structure? Or what's worse is it's the other way around. Someone writes down, oh, I know what that is, the nuclear envelope, and you write down nuclear envelope. And heck, you may write down the sentence, this is the nuclear envelope. It is made up of 4 layers of phospholipids that are arranged in 2 bilayers. All of that is 100% exactly correct information. But if my question was, identify the organelle, while you gave me a ton of correct information, did you answer the question?
No.
No. And if you don't answer the question, I can't give you full credit for it. So again, the key is to make sure that you read the questions carefully. I work very hard. I try very hard to be very specific, very precise in my wording, because as I said before, I love that this class is hard. I don't have to be tricky. I will be very straightforward with you and exactly what it is that I want. You just have to make sure that you give me that. Okay, so I put a lot of time and effort into trying to be very precise so that there's no ambiguity. You know what is the piece of information that I'm looking for. So I try very hard on that. I put a lot of time and effort into the wording of these things to try to make it straightforward, because I don't want the process to get in the way of you showing me I have the information. Yes.
Yeah, so you know, like right now how you're writing, you know, organelle structure, and you know, you're telling us, you know, what the answer is to that.
Yep.
On the practice before the final, or any material that you give us, because sometimes, like, maybe I'm looking in the wrong place, but I don't see all this information through the slides and in the book and all that. So I have no way of studying from, you know, for it, unless I'm looking in the wrong place. So is this gonna be like, are you gonna have a breakdown of each, you know, organelle? You're gonna say organelle structure, you know, how are they arranged, or, you know, you're gonna have that in the answers for us to study?
No. I mean, so yes and no. When we get to things that are more ambiguous, like for instance, we get to the tissues, I will give, if you've looked ahead at our tissue handout, not only will you see on that a list of all the things that you're responsible for, but next to all the things that you're responsible for is a phrase in parentheses. And that phrase in parentheses will be the specific thing, the specific keyword I will use to help you to identify that. As far as the, you know, the cell, every single thing that you see in the cell in front of you, you're responsible for. So it's redundant for me to make a list. What we are doing right now together in lecture is going through and identifying these things and how you need to be able to recognize them. So this is why you take notes during lecture. This is why you can go back and listen to the lecture again a second time to make sure that you get all the information, right? Like I said, if I just gave you a list of all the random things that you needed to go with that kind of precision, then there'd be no point in lecture. And like I said, that'd be awesome. I'd be able to sleep in. But instead, we are here working together to make sure that you've mastered this information. So that's the point of lecture. Lecture is when I'm providing you with this information that's going to help you to be successful. So that's why you take the notes. That's why I'm recording this, so you can go back and to review it so that you know how to be successful in this class.
Okay.
Okay. Trust me, if we stay online for very long, and eventually once I get all these lectures, then I'm just going to let people listen to lectures, and I will be sleeping at that point. If we're doing this again next year, then yeah, I will be sleeping and you guys will just watch lectures, but you'll have gotten the good part at least. All right. Questions on that? All right. So again, these are the pictures of the models from the classroom. I guarantee they'll be on the exam. But again, remember, the goal isn't to just memorize those. I can pick some random pictures you've never seen before and use those as well, as long as they are what I believe to be obvious examples. And I will show you some examples of non-obvious. Well, heck, we already have, right? Clearly, I'm not going to use this for the centrosome matrix because it doesn't have one, right? We'll talk about how hideous this picture is in just a minute when we talk about that a little bit later. All right? Questions on that? All right, let's talk about our next non-membranous organelle. And those are actually extensions that come off the top of our cell. We keep drawing this cell as a circle, but this circle can have structures that extend out off the surface of them. These extensions of the plasma membrane that are formed by proteins and other things inside are other examples of organelles. And there are primarily 3 of these extensions. The first are long hair-like extensions that are known as cilia. The second are long hair-like extensions that are known as microvilli. Well, if both of these are hair-like extensions that stick off, and again, I'll cheat and draw an even simpler example of this. If both cilia and microvilli are long hair-like extensions, finger-like extensions that stick off the surface of the cell, what's the difference?
One moves, one doesn't.
Bingo. One of these are motile, one of these move in a wave-like fashion, and the other are non-motile. They are static, unmoving. The moving ones are cilia. Again, this is another great example of my teaching style. As I mentioned, my goal is to highlight the things that I think are most important and things we don't talk about that are in your textbook you are not responsible for. And this is a great example. Your textbook actually does a really excellent job of describing the 7 steps in the mechanism of how a cilia actually moves in a wave-like motion. It is a really interesting process. I guess it's somewhat useful, but we have so much other material to cover. You're not responsible for it on the exam. If you're interested in knowledge for knowledge's sake, which is sometimes fun, by all means, feel free to go to the textbook and read how the cilia actually move. But it's not going to be on the exam. What is important is to know that they do move and why they move. These cilia, again, move in a wave-like fashion, not to move the cell through space. Their job is actually to move substances along the surface of them. And the classic example of this is right here in your nose. Inside your nose is constantly making mucus. Right? That mucus is capturing dust and debris. It is moistening the air that you are breathing in. Remember, we said the mouth wasn't part of the respiratory system because it's not specialized the same way the nose is. One of the specializations of the nose is this mucus making and these cilia. So it's constantly making that mucus, and those cilia are taking that mucus and moving it to the back of the throat where you can either swallow it or you can spit it out. Now, these cilia are delicate, and they do a good job as long as you're just making a normal amount of mucus. But what if, again, back in ancient times when people were allowed to see other people, you had to be in school and your kid had to go into daycare? The first year your kid is in a daycare, they literally get every single virus that has ever been on the surface of the planet. And they spend that entire first year with their nose running like a faucet. Why is their nose running like a faucet? Because those germs, those viruses that they've been getting cause an overproduction of mucus. And when you're overproducing mucus, that cilia can't keep up with it anymore, right? Back in ancient times, they used to play a sport called baseball. And in really ancient times, they used to play it at a place called Candlestick Park, which was right out on the ocean. And even though it was the middle of summer in San Francisco, when you were out there at 7 p.m. at night, 8 p.m. at night, about the 7th inning of a baseball game, it is freezing cold. And when you're out in that cold for a prolonged period of time, cold slow down the cilia. And as they slow down the cilia, they're not as efficient at moving the mucus. And so you get your nose gets a little runny when you're in the cold for a long period of time. Or my favorite example, there you are at that Justin Bieber concert, and he baby O's, and you lose it. Tears streaming down your face. You're constantly producing tears. Those tears drain down your lacrimal duct into your nasal cavity where it mixes with the mucus. And as long as you're producing a normal amount of tears, those little cilia can keep up with them. But if Justin Bieber's baby-owing and you lose it, you're overproducing tears. Those tears enter into your nasal cavity. Those poor little cilia can't keep up with it. And not only do you have tears streaming down your face, you have snot coming out your nose. All right, but don't worry, Justin still loves you. All right, so those job of those cilia are to move substances. Okay, so then if the microvilli are non-motile and they just stick up on the surface, what is their function?
Capture.
What do you mean by capture? I like that answer. That's a good answer. But what do you mean? Can you be more specific?
I don't know. Capture food.
Ah, I see what you're saying. No, not exactly. The cilia, the mucus that is produced can sometimes help that. What's the advantage of having no non-motile extensions off the top of a cell?
Is
Right,
it
here's my hand.
This is the surface area.
There you go, exactly. Those are the magic terms. If this is a cell, for instance, a cell in my small intestine where I need to absorb food, and this is the surface, I only have this surface area to be able to absorb food. But if I put all these finger-like extensions out, suddenly I have a massive surface area that I'm able to absorb materials from or secrete materials. So absolutely, microvilli are going to increase the surface area. Right? In some instances, that can be used to capture things, but mostly it's about increasing surface area for absorption of things. Excellent. So both of these are finger-like extensions. One's motile to move substances, and one is about surface area for the absorption of substances. All right. But there is a third. The third extension is a flagellum. Flagellum singular, flagella plural. The flagellum is a long singular extension that comes off of a cell, and it actually is connected to a motor. And what that motor does is it rotates. Where's my pencil? It rotates. And as it rotates in a circle, it whips that flagellum in kind of a circular whip-like fashion. And this is actually used to propel the cell in space, right? And in fact, there is only one human cell that has a flagellum. And of course, what is that one human cell?
Sperm cell.
All right, the spermatozoa, absolutely. The sperm cells are the only ones that have the flagellum, and they're used to propel themselves through space so they can work their way through the female's reproductive tract to get to that egg and fertilize it. All right, here we've done it with the words. Let's do it with the pretty pictures. Here are some examples of the cilia, and as I mentioned, your book does a great job of talking about that 7-step process of how they do it. You're not responsible for that process, but if you're interested, you're welcome to read it. And again, their goal of that wave-like motion is to move that mucus along the surface. Here we see an example of one of the absorptive cells in our small intestine with these massive number of microvilli that increase the surface area over 100-fold. Of these. In fact, the small intestine has 3 specializations that increase the surface area of your small intestine over 1,000 times what it would be if it was just a smooth, hollow tube. So it allows us to absorb massive amounts of nutrients from our digestive system. And here is our illustration of a spermatozoon with that nice, long, huge, massive tail. And again, notice they've decreased the size because if you think of the sperm, the head is about 5 microns in length. And the tail is 11 times that, 55 microns in length. So it's basically a tiny little tail, a tiny little cell with a massive tail, that flagellum on it off of the back. All right. Excellent. So let's talk then about, so any questions on those?
Don't the villi in the intestine have microvilli on them?
Yes. So like I said, there are actually three specializations to the small intestine. There are folds in the wall of the small intestine. There are finger-like extensions of the small intestine. And then on top of it are the cells that have those microvilli on top of it. Yes.
Yeah. Wow.
Yeah. Again, over almost 1,000 times the increase in surface area.
The efficiency.
Exactly. All right. Excellent. Our last non-membranous. Again, we are still on non-membranous. Oops. Organelles are the cytoskeleton. Now, again, we already talked about the skeletal system in our bodies. And what were the three main things that our skeletal system, we said, did for us?
Structure, support.
Structure, right? Support or protection. What was the third?
I mean, they make red blood cells and white blood cells, don't they?
True, inside of them. Yep, that is one of those. But that wasn't one of the big three we talked about. You're right. Our skeletal system actually has 6 functions when we get to the skeletal system. But what were the big three? Structure. I heard it. Say it again.
Movement.
Movement. There you go. Excellent. It provided structure, it provided support and protection, and it allows for movement.
Is this
Well,
for non-membranous?
so what I was talking about here is our skeletal system. Our skeletal system, our organ system in our body, our bones provide structure, provide support and protection, and allow for movement. Okay? Now, our cytoskeleton, as it turns out, is a non-membranous organelle that does those exact same things for the cell. It helps to support and protect the cell. It gives the cell its shape, and it allows for movement, both of the cell and inside of the cell. So the same thing our skeleton does for us, our cytoskeleton does for the cell. All right? With me so far?
Mm-hmm.
Excellent. Now, as it turns out, there are 3 main types of cytoskeletal structures. The first of these are what are known as microfilaments. Microfilaments, and actually, you know what? Let's do this way. We'll come back to the pretty pictures in a second. Whoops. Let's go here first. So the first of our 3 main types, as I mentioned, are what are known as microfilaments. They are our smallest of the cytoskeletal structures. They are also known as actin filaments. And the reason they are known as actin filaments is because they are formed from the protein actin. Actin, as it turns out, is a dimer. Dimer is a fancy word. Any idea what dimer means?
Mm-mm.
Dimer basically means that it is a protein that has 2 subunits. Basically, that is what actin looks like. Actin is 2 globular proteins stuck together. 2 things, it's a dimer. Now, because it's so simple, what we're able to do is take 2 actins and very quickly and easily put them together. And then 3 actins and very easily put them together. And I know my circles are no longer perfect and looking nice, but you get the idea. We are able to very rapidly put these actins together, and I'm just going to cheat and draw a line now, as a big, long filament. And that's basically what a microfilament is. It's these big, long strands of these 2 actins put together.
It was in a helix structure?
It does spin around a little bit, yes, but it's not as relevant to its shape. But yes, absolutely. If you look back, as we look at the picture, you will see that it is spiraling as it goes.
Okay.
And that's certainly not something that I can do artistically. But yeah, absolutely. The point of it is that it is pretty simple to put this together. And the advantage of that is it is going to allow for dynamic movements of the cell. Of course, the classic example of it is this one right here, flexing your muscles. When we move our muscles through space, actin is one of the main contractile proteins that allows us to move our body through space. But another really cool example of it is, remember I talked about those nerve cells that are over 1 meter long. Well, when that cell was growing, what happens is it starts in the small of your back with your cell body. And it has to grow this big, long process known as an axon. And the way it does it is by making this growth cone. It makes this growth cone with all these actin filaments, these microfilaments that reach out into its environment. And what happens is some of them reach out to an environment where there are chemical signals that it is repulsed by, and those will dynamically break down. Others will find an attractive chemical signal, and they will expand rapidly in that area. And so by having this growth cone reach out into space, it is able to grow that entire meter of length from the small of your back all the way down to your big toe. And so we get this dynamic movement of the cell that these actin filaments are going to allow. All right. Our second cytoskeletal structure is what are known as intermediate filaments. These intermediate filaments, unlike microfilaments, can be formed by several different types of proteins. So there isn't just one protein that can form them. And intermediate filaments, and again, because they're I for intermediate, I always think of them in terms of the I beams of the cell, right? Back in ancient times, you used to be able to go downtown in Sacramento and walk around. And as you would walk around Sacramento, you'd look up and you'd see these huge, massive skyscrapers. And these huge, massive skyscrapers would be nothing but a complete wall of glass. Now, as you look up at that skyscraper and see that big, huge wall of glass, is it really the glass that is holding that up?
No.
Yeah.
No, of course not. You know that the glass is just superficial. It's on the outer surface. It's decorative. And you know deep inside there is concrete and these big, huge structural I-beams that is giving that skyscraper its shape and its structure. And that's basically what our intermediate filaments do. They are the primary ones that are involved in forming the shape of the cell and the tissues. One of the cool things about some of these intermediate filaments is they can actually extend from one cell to the next, anchoring cells together. So not only do they help to give the cell its shape, but they can give support to the cells next to them as well. All right. And that brings us to our third, the largest, our microtubules. Our microtubules are comprised of, or formed, let's be consistent, from the protein tubulin. Right, microtubules, we've kind of already talked about and hinted at what they do because we were talking about these with the centrioles. So microtubules are made from the protein tubulin. These microtubules are basically the highways and byways of the cell, right? What that means is they allow movement inside the cell. Think of it this way. Back in ancient times, and again, I have to work on my analogies because not all of them work anymore. Back in ancient times, we were on campus right now. And I would remind you that most of you got here today because of a car, and that car drove on the highway, right? You used Interstate 80 to get to campus, And did Interstate 80 move at all to bring you to school?
Negatory.
Yeah, the road doesn't move. It just forms the pathway along which you traveled. And that's what microtubules do. Microtubules don't actually physically move things. What they do is they are the pathways that other things can move, right? They are this long highway inside of the cell. And inside the cell, we have these very cool things that I think we already talked about once, but I'll remind you of them again, called motors. Remind me again what a motor does?
Allows movement through space.
True, it produces a mechanical force or mechanical energy, that movement through space. But what does it use to produce that mechanical energy? What does it use to make that mechanical energy?
ATP?
Right, which is a chemical energy. Excellent. Use a chemical energy to produce a mechanical energy. And that's exactly what these motors do. These motors basically have little legs. All right, hold on. Let me draw that. Doot, doot. And it has these little legs, and what it does is it uses an ATP to produce a mechanical force, and it uses that ATP to basically walk along the surface of the microtubules. We talked about how these microtubules are going to help to move the chromosomes around when the cell divides, but we can also move other organelles or other structures along inside of the cell. So notice they're not changing the shape of the cell, but they're allowing movement to take place inside. So these are our three main types of cytoskeletal structures. And now that I've written this out, let's go back to the pretty pictures from your textbook, and we can see what this looks like. So again, here we see some cells with those actin subunits, again, those dimers. And again, you can see it is helical in its nature, it is spiral in its nature throughout. And they allow for dynamic movement. All of these support, all of these produce the structure, but microfilaments also can allow for dynamic movement of the cell. Our intermediate filaments, again, give shape and structure to them, right? These are the I-beams that give us that structure and support. And here is our microtubule. And again, these are the highways and byways that allow movement inside of the cell. Here we see them individually, and here we see the pretty picture from the textbook that helps to show you some of these. Notice our microfilaments are helping to form the structure of our microvilli. Our intermediate filaments are giving the shape to it, and notice they're also attaching to this structure that's going to hold to another cell next to it. And then notice here I have my microtubule where I have a vesicle or I have a little bit of an organelle that are moving along the surface of that highway and that byway. So all of these give the shape and structure, but they're also unique in their own way with some specific functions to them.
So what is moving through the microtubules?
Nothing. I mean, there really isn't anything that moves inside of them. So it's more, it's like the highway. They move, things move along the outer surface.
Oh, okay.
Yep. Yeah, so they don't move through them. I apologize.
So then what moves along it then?
Like motors, like we talked about. We can have those motors. So this vesicle is attached to a motor and it is being moved to a different part of the cell, right? So like I said, a great example again goes back to those nerves. If that nerve is going to connect from your small of your back to your big toe, so that when you step on something sharp, you move that toe away, that nerve needs to send a chemical signal to the cells way down at your big toe. But that chemical has to be made back in the cell body. So what happens is there's some microtubules that run the entire length of that axon. We attach the chemical in a vesicle to it, and it gets transported all the way down to the cell, to the end of the cell, so that it can be used.
Okay, gotcha.
Yep.
Professor, I have a question.
Yes.
Okay, so just to see if I got it correctly. So the microtubulus, it allows the movement inside the cell. It's
Correct.
like a pathway, right?
Yes.
And connected to it is the motor to...
Yes, so the same way that your car moves along a highway,
Okay.
Motors can move along the surface of a microtubule.
Okay. Got it. Thank you.
Yep.
And they generally transport chemical signals.
Or like I
Chemical
said,
med
remember,
senders.
when the cell divides, we have to move the chromosomes around inside the cell.
Okay.
It's going to be the microtubules that do that.
All right.
So anything that needs to be moved around inside the cell, the microtubules are going to help to do it. Okay. Awesome. All right, that is our last non-membranous organelle. Let's talk some about our membranous organelles. Obviously, again, these are the ones that are bound by membranes. And here is our first classic example of this. This is our endoplasmic reticulum. Now, again, remember, as I've mentioned, spelling does matter in this class. So on the exam, if I had a big arrow pointing to this and you just wrote ER, you're only getting partial credit. That is considered a spelling error. You need to spell out endoplasmic reticulum. All right. Now, if on an essay question I asked you to describe what the smooth or rough endoplasmic reticulum did, once you wrote that out once, you could then abbreviate things ER after that. So that is totally acceptable. But remember, spelling matters, and so do not abbreviate on the exams, especially the lab exams. All right. Now, again, the same way that our centrosome is an organelle that is comprised of structures, our endoplasmic reticulum is comprised of structures as well. If you notice, basically what we have here are a bunch of flattened membranous, or let's say it this way, flattened interconnected membranous sacs. And because our anatomists love to name everything, they give a specific name to these. And the name for this is a cisterna. Cisterna being the singular, cisternae, with the E being the plural. Now, as I mentioned, spelling does matter. And I'm not sure if I talked about this during the initial introduction to the class. Spelling does matter, but I will give you one letter. So if you put an A here instead of an E, or maybe you transpose the E and the R, I will give you one of those. So again, a minor spelling error like that, you will still get full credit. But if you get more than one wrong, then you will lose partial credit for things, because spelling does matter. Notice there's only one letter difference between the singular cisterna and the plural cisternae. So technically you could write it either way and it would still be correct. So again, while it will eventually be important for you to be able to distinguish singular and plural, if you write it either way, I will be happy with that. But what we have are these interconnected flattened membranous sacs known as the cisterna. All right. Now, and again, I think I actually have that right there. There you go. Cisternae, plural, cisterna, singular. Excellent. However, if you noticed, the cisterna come in two different flavors. Some of them, like these here, have these globular structures attached to the outer surface. These globular structures attached to the outer surface are what are known as ribosomes. These make these flattened membranous sacs look much more bumpy, much more irregular in their appearance. And because of that, these cisternae that have the ribosomes on it aren't just endoplasmic reticulum, but what kind of endoplasmic reticulum are they?
Rough.
Rough.
Yeah, they are the rough endoplasmic reticulum. Excellent. Oops. I get one letter just like you guys. There you go. Excellent. Now, function. What do these ribosomes do? Anybody know?
They process DNA to make proteins.
Close. You are absolutely correct. They do indeed, the ribosomes make proteins. However, remember the DNA is locked up inside the nucleus. The DNA doesn't get to come out and play. However,
RNA?
there you go. We can make a temporary portable copy of the DNA And so ribosomes use RNA to make proteins, absolutely. And notice, when you look at the anatomy of this in your textbook, notice, as you can see, the rough endoplasmic reticulum is typically right next to, and sometimes will even attach to, the nuclear envelope. So that as our RNA comes out of it, the first place it goes is,
It's just
boom,
ready to go.
right to the rough endoplasmic reticulum. Now notice, and again, this is where our artist has let us down a little bit, not all the ribosomes are going to be bound to the cisternae of the rough endoplasmic reticulum. Notice when we look here, these look like they're bound to the nuclear envelope, don't they? They're not. But there are some free-floating ribosomes. So most ribosomes are bound to the rough endoplasmic reticulum, but not all of them. There are some free-floating ones that you will find just circulating within the cytoplasm of the cell. All right? Now, notice these cisternae over here don't have ribosomes on them, so their appearance is much more uniform, or what we would call, in this case, the smooth. And because I'm running out of space, and I've already written it out once, I will abbreviate it ER for the smooth ER. Now, the fact that it doesn't have ribosomes, what do we know the smooth ER doesn't do? What can't it do?
Make protein?
It can't make proteins, absolutely. The smooth endoplasmic reticulum cannot make proteins. So what does it do? Anyone know?
It provides support.
Not a bad guess. It does provide some support, and one of the ways it does that is by actually making lipids. It is actually able to play a role in lipid formation, so it's going to form lipids and things along those lines. It also is able to break down and detoxify some lipid-based materials inside of it, so it can help to recycle materials that way. And as we'll see in some cells, the smooth ER can actually be specialized for storage. For instance, in our muscle cell, where we have this elaborate process of a muscle contraction, the smooth ER is actually specialized for storing calcium, because as we know, calcium makes cells do wonky things. It can metabolize lipids, it can form new lipids, and it can store materials.
Yeah.
Now, let's cheat. Hold on. Here, all the way back to here. Again, remember, as we mentioned, most rough endoplasmic reticulum is in close proximity or attached to our nuclear envelope. Is that the case here?
No.
No, right? What is this here? Identify this organelle. Oh, where'd my arrow go? Hold on, let me make an arrow. My arrow disappeared. There we go. Identify the organelle.
Endoplasmic reticulum.
And you get partial credit for that, but that's not specific enough. Be more specific.
Is that the smooth?
Smooth endoplasmic reticulum. What organelle is that?
Rough.
Rough endoplasmic reticulum.
Yeah, excellent. That is the rough. Now notice I could keep that same pointer in the same area and ask you to identify the structure. Identify the flattened structure, and your answer would be?
Cisternae.
Cisternae. I can move it slightly and ask you to identify the globular structure. What would that?
Ribosomes.
Ribosome. And what's that? Identify that globular structure.
Ribosomes.
These are ribosomes. Remember, we talked about some ribosomes are free-floating. Now, are they free-floating in schools, like schools of fishes clustered together in one location?
No. Not.
No, absolutely not, right? But again, we have, and again, we won't say the artist is wrong. We'll just say they took a little bit of artistic license with this, right? But clearly these are the red dots that are similar to these red dots up here. So I think it's pretty obvious that they're supposed to be ribosomes. These flattened interconnected membranous sacs have these globs on it and these don't. So even though their locations aren't exactly perfect, they're still obvious where we can tell what they are. These are obvious examples of these things, right? Is this smooth or rough? Right here.
None.
Mm,
Oh,
This
those.
smooth, yeah, not the
it's smooth.
arrow, my highlighter. You guys can
Yeah.
see the highlighter, right?
Yeah.
Smooth or rough?
Smooth.
Smooth or rough?
Rough.
Rough.
Smooth or rough?
yeah.
Smooth.
Smooth or rough?
Rough.
Rough.
Excellent. Now remember, there was another picture too. Let's take a look at that one. Notice here, they've done a good job of at least starting in the right place, where we have flattened membranous sacs with these white dots on them that are actually surrounding and attached to the nuclear envelope. This is clearly what organelle?
Rough endoplasmic reticulum.
Rough endoplasmic reticulum, right? Now, when we look a little closer, one of the things we may notice is that they have this rough endoplasmic reticulum, actually these interconnected sacs, connecting to the outside of the plasma membrane, right? Notice down here, it looks like a slip and slide that goes all the way outside of the cell. Is that what our rough endoplasmic reticulum does?
No.
No, of course not. And notice, this one has dots, this one has dots, these have dots, these have dots, these have dots, these have dots,
Yeah,
this
where's the
has
smooth?
dots, this have dots. Yeah, where's the smooth, right? If you look really closely, and I will highlight it, this one right here is the only bluish membranous sac that doesn't have white dots on the outer surface. And it's this bulb connecting to the outside of the plasma membrane. I'm guessing this is supposed to be our artist's representation of the smooth endoplasmic reticulum, but have they done a very good job of it?
Negatory.
No. So is this the example that I'm going to use to represent the smooth endoplasmic reticulum on the exam?
I'd hope not.
No, absolutely not. Like I said, I don't have to be tricky. I don't have to be, challenging. I'm not going to use something stupid like this because I will use obvious examples. Doesn't mean that there isn't good stuff, right? They have a very nice nuclear envelope. They have very nice nuclear pores. What would this structure be? That red dot
Nuclei.
right there. Any idea what that would be?
The nuclei or nucleus?
There you go. Well, but remember, it's just one though, so it would be the nucleolus.
Oh, yeah.
Although, like I said, if you put nucleoli on this exam, especially for the first exam, even though you use the plural, I would still give you full credit for it, right? Notice our previous model does a little bit of a better job. This is a better one. Notice we have the loose red material in here. What would this substance be?
The chromatin.
Chromatin. And these gray dots, what do they represent?
The nuclei.
The nucleoli, remember? Nuclei is the plural of nucleus. These are nucleoli,
Yeah, sorry.
right? Or one would be a nucleolus. So they've got chromatin, they've got a nucleolus. This is the one that had the matrix. So again, every model has some things that are good, some things that are bad. But man, the smooth and rough endoplasmic reticulum on this model is really, really bad. Yes, Ryan?
So how much variance is there between cells and the amount of organelles that they have?
It can be massive. And again, so that's the goal. What we're looking at right now is what a typical cell looks like, right? And that model that we have, that is a quote unquote typical cell. And a lot of the cells in our body look like that, but most of the cells of the body are also going to have something special about it. Remember, as we talked about, they're specialized. So when we get to, for instance, the muscular system, we will see that muscular cells are very, very different. Right? When we get to blood, we'll learn about how blood cells, and again, that won't be till 431, are very, very different. So some cells are more similar, some cells are highly, highly specialized. But we need to know the basic building block first before we can start talking about the specializations. Because once we see what a typical cell looks like, then we can start saying how these other cells are different from that.
Yeah, I think the only one I know is heart muscle might have more mitochondria.
A lot more mitochondria, absolutely. But it also has a lot of special organelles that help it to contract, too.
Okay.
Yeah, we'll get to that, too, when we get to the muscular system. All right? Excellent. All right, so those are the rough and smooth endoplasmic reticulum. And we've identified them, the components that make them up. Again, the table tops and the table legs that make the table. So we have organelles and we have structures. and then we also know their functions. Let's talk about another important organelle, and that is our Golgi complex. Remember, when we talked about proteins, we talked about the levels of organization, right? Primary, secondary, tertiary, quaternary, those 4 levels of organization for our proteins. Our DNA and the RNA produce basically the first level. That gives us the sequence of amino acids that we're going to put together. And that's really what our rough endoplasmic reticulum does. It makes the strand of amino acids. That strand of amino acids, once it's made, comes to the Golgi complex. And what the Golgi complex is basically responsible for is it cuts and folds the polypeptides to give them their tertiary shape, and then it puts the polypeptide segments together to make the functional protein, right? Remember, I used the example in the introductory class that a cell is a factory, right? You have this factory in China who's responsible for making all the shoes for Nike. So you have all those workers on the floor of the building who are making the shoes, and those are the ribosomes. The ribosomes are the workers who make the shoes. Once they make the shoes, they throw those shoes into a pile, and it's somebody else's job to make sure that they have a left shoe and a right shoe of the same style, that they're the same sizes, and then they put them in the box and package them up for shipping. And that's what our Golgi apparatus does. Not only does it cut and fold the polypeptides, not only does it make the functional proteins, but once it makes the functional proteins, and I've run out of space up here, so I guess I got to cheat and move my writing down here. Once it makes the functional proteins, then it ships the proteins to their final destination. Right? That is the function. That is the function of the Golgi apparatus, or what is also known as the Golgi complex. All right. We started first with the function this time. Yes, Ryan.
How does it know where to ship?
How is one of those challenging questions. These are things that are can be difficult for us to study and understand. How it actually knows where to do it isn't fully understood, but we do know that different proteins are going to go to different locations and do different things. But how it actually knows to do that, my guess is, again, it's probably more chemical signals that are taking place inside of it. But again, these are things that are really hard to challenging to study at a molecular level. To watch cells function at a molecular level in real time is something that is very challenging. So you're asking some of the questions that we don't necessarily know the answer to. That's one of the challenges with anatomy and physiology. A&P, right? The architect who put this together, all of this together, didn't leave us her footnotes. So why is one of the most challenging questions to be able to answer. We can often infer how some of these whys are occurring or why things happen, but there's a lot of basic stuff about anatomy that we still don't fully understand, especially at the cellular and molecular level.
And then one more. What are the vesicles made of?
I'm sorry?
What are the vesicles made of?
Oh, that's where I'm getting right now. Let's talk about
okay.
Okay? So what happens is that rough endoplasmic reticulum, as you can see here, is going to pinch off a little bit of its plasma membrane. And when it pinches off a little bit of its plasma membrane, it's going to have the proteins either inside of it. So we could have proteins that are inside of it, or some of the proteins will actually be, I got to change colors for this one, embedded within the wall of the plasma membrane. This structure, a plasma membrane with proteins inside and or outside, we have a fancy word for, because we have a fancy word for everything, and it is a vesicle. So notice this vesicle comes to the beginning part of our Golgi apparatus. We haven't talked about the anatomy of our Golgi apparatus yet, but our Golgi apparatus is also made of flattened membranous sacs. And what was the term we had for flattened membranous sacs?
Cisterna.
Cisterna. Exactly, cisterna, or again, plural, cisternae, are those flattened membranous sacs. However, these are not interconnected. So in this case, what we have are these flattened stacks that are like stacks of pancakes right next to each other, but they are not interconnected. And what happens is the vesicles from the rough endoplasmic reticulum, and I'm going to abbreviate for speed, enter into one side and then basically get passed by vesicles back and forth between the layers. And as they do, they get processed. And at the end, when they're done, those proteins are then bundled into a vesicle that then leaves the Golgi apparatus and goes to whatever its final destination is. And as we'll see, there are basically three potential outcomes. And we'll see those in just a minute. All right, but first, again, I've kind of drawn a silly illustration of this. Let's look at a better example. So again, it's about processing, delivering proteins and lipids. It allows us to modify the plasma membrane. We'll see how that works in just a second, but this is the picture I wanted to show you. Notice this is how this process works. Notice there are proteins inside the cisterna. There are proteins embedded within the plasma membrane of the cisterna, and a small portion of that plasma membrane pinches off, buds off. And when it does, it becomes a vesicle. So as you can see, a vesicle is just a little bit of plasma membrane that it can have proteins inside of it, in its wall, or both. It then comes to the next cisternae, where it fuses with it. Notice the plasma proteins that were on the surface become part of the surface of our cisterna, and those inside are released inside. And they can be cut, and they can be modified, and they can be folded, and they can be put together. These proteins move through the Golgi apparatus in one direction. Notice on the models, it's usually pretty easy to find the Golgi apparatus because, again, we're looking at anatomy. And when we're looking at that anatomy, there are these flattened membranous sacs with lots of vesicles associated with them. But one side always faces the rough endoplasmic reticulum, and one side always faces the plasma membrane because our proteins only go in through one direction. They enter at the starting phase, right? And that starting phase is also what is known as the forming phase or the cis phase. And it comes out the maturing or what is known as trans phase. So our proteins only go one way through the Golgi apparatus. All right. Now again, if we cheat, and this will be easiest if I actually stop this for a second. Come back here, and not that one, this one. Oops, no, wait, oh no. All right, I gotta cheat and go all the way back. Oh no, actually I don't want to go that way, I want to go all the other way. There we go, oh no. All right, hold on, let's try this one more time. Pretend I know what I'm doing. There you go, that's what I wanted to do. Oh, that's the problem, time for another break. All right, hold on, before we do that, Excellent, all right, so what color is our Golgi apparatus on this particular model?
Red.
Yeah, red, exactly. Notice it is, but I need to bring up my annotation tools again. Here we see those flattened, connected, one side faces what should be the rough endoplasmic reticulum, but at least it's facing the nucleus where we know things are going to come out. and they're going to come out. So this is the cis phase, or the forming, maturing, coming out. And notice they even have some vesicles coming out of the maturing end, right? Notice this is the actual model that we have in the classroom. Again, notice we can see this one a little more clearly with those dots of the ribosomes, and that's the only one that doesn't have it. What color is our Golgi apparatus in this one?
Purplish or pinkish?
Yeah, obviously we can see here our cisternae, and we have some vesicles, but notice they're interconnected to each other and for some reason connected to the nucleus. So this is a horrible, horrible example. This is the one we actually have in the classroom. They did actually update the model. Notice when they updated the model, they did a much better job of showing us an appropriate Golgi apparatus. So now this is an appropriate Golgi apparatus, but notice they didn't bother to fix any of those rough endoplasmic reticulum. So this is still a pretty bad model, but at least they improved the Golgi apparatus really, really nicely. So again, we have those flattened cisternae, we have those vesicles associated with them there. All right. Questions on that?
The vesicles never really leave the cell then, huh?
Yes and no. So that's actually the last thing that I wanted to talk about for this. So let's switch back to the whiteboard so we can talk about this. All right, so let's go ahead and draw the plasma membrane. And let's go ahead and draw our last cisternae of the Golgi apparatus. All right, so this is our Golgi complex or Golgi apparatus. So just, again, I'm going to abbreviate for simplicity here. As we talked about, there are basically, once we make that functional protein, as we said, our Golgi apparatus is going to send that protein to its final destination. And there's basically 3 possible paths for the proteins. All right. And so let's identify and see what those are. Part of those paths are going to involve where the protein is. So let's take a look at vesicle 1. In this particular vesicle, this vesicle has proteins on the inside. Okay? So what we're going to do is draw our vesicle. And on the inside, what does that look like? Oh, no, I don't like that. We will put 4 proteins on the inside of this. All right? So this is a vesicle that has proteins on the inside. What's going to happen to this vesicle is this vesicle is going to migrate to the plasma membrane. When it migrates to the plasma membrane, what it is going to actually do is, oh, that, I don't know what I just did there. I don't like that. Sorry, trying to move things around so I can see what I'm doing. And great question, Madison. Golgi apparatus, Golgi complex are the exact same thing. They're the same terms for the same thing. Golgi apparatus, Golgi complex, 2 names, the same organelle. So they're both appropriate anatomical terms for the organelle. All right. With vesicle 1, the proteins are in the inside. And basically what happens is the vesicle comes to the plasma membrane. And when it comes to the plasma membrane, and actually I'm going to draw this, it fuses to the plasma membrane. Fuses and becomes a part of the plasma membrane. And when it does that, what's going to happen to the proteins that are inside of it?
Comes out?
Yep. Oh, so hold on. Shoot. Thought I was trying to be clever here. Hold on. Let's do this one more time. That, like that. Excellent. But then I have to find my words and turn those back to black. Oh, and I can't do that. All right. I have to rewrite that. Actually, let's just cheat and go back this way. Oh, well, let me go back. All right. So again, fuse this to the plasma membrane. And as you guys mentioned, when it does that, it is going to release the proteins out of the cell. All right, so those proteins that were inside of it, that were purple, are released from the cell. And we have a fancy name for that process. What's the fancy name for that process?
Exocytosis.
There you go, exocytosis. So basically, option 1 for the proteins are to be released from the cell via exocytosis. That's one possibility of what can happen to the proteins. Not sure why that's not all feasible. Okay, we comfortable with that idea? Stunned silence means we
Yes.
totally understand it, Dr. Slutsky, and so I continue to move forward. Excellent. Option 2 is where the proteins are embedded in the plasma membrane. So in this case, we have a vesicle, and the proteins are on the plasma membrane. Once again, this vesicle is going to come to the plasma membrane. Once again, it is going to bind to the surface of the plasma membrane. And I will do that again. However, notice this time the proteins aren't going to be released from the cell. Instead, what happens is these proteins that were embedded in the plasma membrane of the vesicle are now basically going to be on the surface of the plasma membrane of the cell. So basically what's happening here is these proteins are expressed on the surface of the cell. So this could be, for instance, a new receptor allowing, or maybe a channel that allows sodium to come in, or a pump that kicks out calcium, or maybe it's a receptor for a hormone to bind it and influence the cell. Notice by doing this, by putting these proteins on the surface of the cell, we can change the plasma membrane. We can change the work that it does. And that's a pretty powerful thing, to be able to change what a cell is doing by putting new proteins on its surface. All right, so far so good?
So is that a part of differentiation?
It is one of the things that can make cells different. Do all cells express the exact same proteins on their surface? No.
No.
So absolutely, so different cells will be expressing different proteins, absolutely. And this is one of the ways that different cells can have different jobs. Now, the third thing that can happen, and again for this one, with the vesicle, protein, oh, so I spell it right. It doesn't matter, proteins can be on the surface or inside, it doesn't matter for this one. Because what's special and different about this instance is that this vesicle does not bind to the plasma membrane. Instead, oh, that's not how you spell not. Instead, basically, it stays inside the cell. And if you think about it, it is a piece of membrane that is going to have proteins either on its surface or inside of it. And it's going to have some kind of job. Essentially, what it does is it becomes an organelle. In fact, we have over a dozen different vesicle organelles floating around inside of our cells. Now, while there are dozens of them, there are only two I'm going to hold you responsible for. All right. Now, notice I've done an amazing job of drawing this with the beautiful picture. Truly stunning, right? Hang it on your wall. But let's take a look at the pretty picture from your textbook that shows the same thing. Notice when we come back to this picture, here we see a vesicle that has, come on, grab this and get out of my way. There you go. Vesicle that has proteins on the inside. There's my annotation token. There you go. Highlighter. Proteins on the inside coming to the surface and being released via exocytosis. This one must have proteins on the plasma membrane because it's fusing and now becoming part of the plasma membrane. And this one is becoming a vesicle, and that vesicle is coming and doing some job inside of the cell. Now, as I mentioned, there are over a dozen different vesicles that can be found, vesicle type organelles that can be found inside the cell. But let's talk about the two most common ones, because these are the ones that are going to be most important to us and the ones you'll be responsible for as well. The first are lysosomes. Lysosomes contain hydraulic enzymes. Hydraulic enzymes, does that mean they break bonds or make bonds?
Break.
Break bonds, excellent. Right? I know it's all very popular now to be green, but cells were green before it was hip, right? They are the ultimate and original recyclers, right? I have two children. I think I've mentioned this, two daughters. Especially when they were younger, they loved Legos. So they would take all their Legos and they'd build a huge, massive house out of all of their Legos. And once they got done building the house, then they wanted to build a boat. So what did I do? Do I go running out and buy them more Legos?
No.
No, I would grab the house, smash it on the floor into a bunch of little pieces, and say, there you go, now you've recycled them, and now you can build your boat. Okay, I never did that. Actually, I went out and bought them. Legos, I'm horrible that way, but you get the idea. There are organelles inside of our body that are no longer being used. They may be damaged or dying. Proteins that aren't being used anymore. Right? We all have those Christmas lights that are taking up space in our garage right now because it's going to be months before we're going to be able to use them, right? Wouldn't it be way more awesome if rather than taking up that garage space, we could break them down into other things that we needed, like electric fans or air conditioners or things like that now, and then once Christmas came, we could reassemble them back into lights, right? Well, our cell doesn't have the storage space that you do, so we don't have the space to keep proteins that aren't functional, to have organelles that aren't working anymore. And so these lysosomes can help to break those things down. There are also some special cells that use their lysosomes to break outside things down. Some of your white blood cells are things we call phagocytes or macrophages. These are cells that are responsible for finding something that doesn't belong in your body, like a virus or a bacteria. And what they do is they endocytose it, they bring it inside of them, and then they use these lysosomes to break down that virus and make it non-functional, destroying it so that it doesn't harm you anymore. So these lysosomes help us to break down both internal and external things that either we don't need, don't want, or could harm us. Now, the second most common type of vesicle organelle is what is known as a peroxisome. Peroxisomes play an important role in detoxifying the cell, especially of hydroxyl groups, which can be very damaging. They also help to break down fatty acid and other type of organic compounds, toxins, pesticides, and things like that that are absorbed into your cells. So these are important cells in helping to clean and detoxify the cell and help to keep the cell healthy in that way, especially like those free radicals and those hydroxyl groups, things along those lines. All right. Now, we do have one minor issue with this. We come back to our pretty picture. And in our pretty picture, we are looking for and to identify our organelles. And both the peroxisomes and the lysosomes are vesicles, right? We could pretty clearly tell the difference between our rough endoplasmic reticulum and our smooth endoplasmic reticulum and the Golgi apparatus by their anatomy. But when it comes to vesicles, they're just kind of ball shaped. And notice, conveniently enough, there are two you are responsible for. And I see two different flavors of ball-shaped vesicles inside of our cell. So the question becomes, which is which? In real life, which one is purple, a lysosome or a peroxisome?
Can you say that again?
In real life, if I were to actually look inside of one of your cells right now, right, obviously some of your vesicles would be purple, some of them would be yellow. Which ones are the purple ones? Is the peroxisomes purple or the lysosomes purple?
The lysosome.
Lysosomes?
Well, of course, neither. Yellow and purple is.
Matter?
No, it wasn't a trick question. There, obviously, this is an artistic coloration of them. There obviously is no real color, so we can't really use color as the real rule of how we do it. However, there are some subtle differences. Now, again, it might be easy from a mnemonic standpoint to think purple starts with P, peroxisome starts with P, so let's just do that. But again, there's that magical phrase, and I guess I'll have to write it in white here. By convention. I think we've talked about this already in class, right?
Mm-hmm.
By convention basically means a rule that has been made up by typically anatomists to decide what is what. Now, in this case, we are going to make a rule by convention for which is which. Now, we're not just going to flip a coin. We are going to use a real rule. And as it turns out, typically, lysosomes are larger than peroxisomes. So in real life, while their colors may not be accurate, lysosomes are larger, well, that should be are larger than the peroxisomes are. So as I look at the purple things and I look at the yellow things, which ones are larger?
Purple.
Purple. So for us, our rule that we are going to make up by convention is that the purple vesicles are going to be our lysosomes, and the yellow are going to be the peroxisomes. All right, so that is the rule we are going to make up. Purple vesicles are going to be the lysosomes, yellow are going to be peroxisomes. And if you notice, luckily, conveniently enough, when we look at the other cell, notice they have two different colors of vesicles as well, and it also happens to be purple and yellow. Now on this one, you could argue that some of the yellows are bigger than the purple and some of the yellows are smaller than purple. Again, they were a little lazy with this model. But since we have a color rule, we are going to stick with that color rule. So this one here, regardless of its size, this one here, regardless of its size, what are these two things?
Lysosomes.
Lysosomes?
Whereas this one here and this one here, these two yellow things are going to be
Peroxisome.
peroxisomes. There you go. Exactly. So that is going to be the rule we are going to use by convention. Okay?
You mean that's going to be the rule for the exams, right?
For the exam, yep. So if I have an arrow pointing to this thing on the exam, asking you to identify this organelle, you're going to say lysosome. If I have an arrow pointing to this thing on the exam, you're going to say peroxisome. Okay?
Okay.
So that is our rule that we're making up for the exam. All right, let's clear that. Let's do that. Go back here. Excellent. And that brings us to our last membrane-bound organelle we are responsible for. It is the mitochondria. Notice our mitochondria is our second double membraned organelle. What was the first one again?
Nucleus.
Nucleus. The nucleus had two membranes, and so does our mitochondria. Although notice our mitochondria's double membrane is slightly different. It has a smooth outer membrane and an elaborately folded inner membrane. Now, notice this is a folded inner membrane, and because anatomists love to name things, they are going to give a name to this folded membrane. Is it a flattened membrane?
No.
No, it is a folded membrane. Flattened membranes are cisternae. Folded membranes are cristae. And fun with vocabulary. Similar words, totally different. We're going to take the break right after this here. Similar terms, totally different. A crista, singular, cristae, plural, are the folded inner membranes of a mitochondria. whereas cisternae are the flattened membranous sacs that make up the Golgi apparatus and the rough endoplasmic reticulum. Now, the reason this cristae is all folded is because it keeps all of the molecules in here close together, because what is it that our mitochondria do?
Oh, make energy.
Produce ATP.
Produce ATP, absolutely. And they do it in a very important way, right? As we talked about, our main fuel is sugar. And basically, in the presence of oxygen, when there's oxygen, our mitochondria are able to completely break down that sugar to produce energy, and that energy is used to make ATP. And as a waste product from this, we also get water. And the other waste product is carbon dioxide. This process of using oxygen to completely break down sugar to get energy, water, and carbon dioxide is what we actually call cellular respiration. All right? That's the whole reason you do this, is to get that oxygen to your cells so they can make ATP and to get rid of that excess CO2. It's the whole reason we breathe, is because of this cellular respiration that is taking place at a cellular level. Now, this process occurs by what is known as the citric acid or Krebs cycle. You'll get to learn about that in micro, so I'm not going to bother teaching you about it. You just need to know what cellular respiration is and know that it takes place here in the mitochondria. The mitochondria has a double membrane like the nucleus. It also shares another characteristic with the nucleus as well. It actually contains its own DNA. Right? When a cell comes time to divide, the cell has to make more Golgi apparatus. It has to make more endoplasmic reticulum. So both new cells gets everything that it needs. But as it turns out, mitochondria actually have their own DNA and are capable of dividing themselves. Now, we get our mitochondria from our mothers. Remember that sperm basically is just a chromosome missile to get the chromosomes to the egg. All of the working parts of the cell come from the mitochondria. Mitochondrial DNA is actually easier to get. So often when, like in the CSI type things, when they're doing, looking at DNA from, skin cells or other things like that that they're using, it's often mitochondrial DNA that they're looking at. However, when Moripovich is trying to decide if you are the father, are they looking at mitochondrial DNA from that?
No.
No, for that they need nuclear DNA, because you only get your mitochondria from your mom, so it wouldn't tell you who the dad was. All right? Questions on that? All right, excellent. With that, again, your book's got a nice table. Make sure you're familiar with that. Make sure you're familiar with examples, pictures, illustrations, models of these things, because that is what you're going to be responsible for on the exam. All right, with that, we did go a little long. I appreciate that, but we still have a fair amount to cover today. So we'll go ahead and take our last break now, and then when we come back from our last break, we are going to do mitosis. All right, so again, we'll take a good break, a 12-minute break, come back at 11:15. At 11:15, we will pick up from here. So any questions before we take a break?
Yeah, quick question. You said about the table. Is the table still in the slides, or is it that
in your textbook? When I refer to tables like that, I'm talking about in your textbook. Your textbook has tables and illustrations that
all of
these things and talk about all these things there.
Okay.
Was there another question too? Yeah.
Yes. So the other lectures, did you post them on Canvas or?
No. So hold on. Let me write this. So
on the YouTubes.
There you go. Yes, when one of the things that I learned over the spring when we were trying to do this is that while I know when you go to the Confer Zoom, there is a place for recordings there. If we let Confer Zoom try to do the recordings, it's not consistent about being able to reliably do that. So what I found was much more reliable is that I record them just rather than doing it in the cloud and letting Confer Zoom do with them. If I record them to my computer, then I know I have them, I can convert them, and then I get them posted onto the YouTube, and then I know it gets there. Confer Zoom was a little faster, but it wasn't consistent. And so I think it's much more important to make sure it gets there, even if it's not till the next day. At least I know with 100% certainty you're going to get the recordings. So if you look in the modules there under the study tools, there is a link to our YouTube channel. And then once you go there, you can actually subscribe to it. Right? Smash that like and subscribe. But all kidding aside, you can actually subscribe to it. And then so that way, when the new lectures post, you will get a notification about it. You don't have to subscribe. I don't care if you subscribe.
Sure.
I'm not interested in being YouTube famous, but at least that way you would know when they come up. But usually, especially with a morning class like this, I can usually get it up and get it because it takes time for me to convert it. It also takes time for me to upload it, and then it takes time for them to process it. So often it will be done by the afternoon, or pardon me, by the evening it will be available. However, depending on what I have going on in real life, I may not be able to get to it till the evening, in which case it's not going to be up till the next morning usually. But usually definitely within 24 hours, usually sooner than that, they should be posted on that YouTube channel. All right?
Mm.
All right, excellent. Let's meet back here. We took a couple extra minutes. Let's go ahead and meet back here at, here, hold on. Let's erase this. And let's restart. What time is it actually? At 11:06. So let's meet back here at 11. We'll go 20. We'll take a little bit of an extra break because that was a lot of material. And mitosis is going to be dense, especially if you're not familiar with it. So we'll come back at 11:20. That'll still give us a little over an hour to do mitosis, which should be enough time for that. All right?
Could I ask you a quick question about the vesicles again?
Yeah, sure.
So the vesicles, you say they fuse to the plasma membrane, right?
That's what this model is showing right over here. Right over here, we can actually see where it's bound to the plasma membrane, becoming part of the plasma membrane, and releasing its contents. Again, if you think about it, if we go back to the picture, the same way that this vesicle is binding to the cisternae and fusing and becoming a part of it, that's exactly what it does to the plasma membrane.
So does that not increase the surface area of the plasma membrane?
Yes, but other parts of and again, cells do get larger, and that's one of the ways we can make the cell larger. But remember, we can also endocytose some of the plasma membrane as well. Our plasma membrane's constantly dynamic. It's going to be constantly dynamic, yes.
Okay, it's like an in and an out for the vesicles.
Exactly.
It's kind of dynamic. Got it.
Yep. But cells also get bigger, so that's definitely one of the ways the cells can get bigger too.
Interesting. Okay, thank you.
Yep. All right. Great questions. Any others? All right, I'll meet you back here. Oh, yeah, go ahead.
Sorry, quick question. Are we going over the mitosis lecture or the cell membrane and transport?
We are going to do mitosis first.
Okay, awesome. Thank
Plasma membrane, we're going to save for tomorrow. Plasma membrane and membrane transport is a big process, so we're going to save that for tomorrow. Not that mitosis isn't a big process, but it's a little bit more straightforward. So we're going to do mitosis today, our big first physiological process, and then plasma membrane and membrane transport is going to be key.
Okay, awesome.
All right.
Thank you.
All right, I will see you guys at 11:20. Let's go ahead and get started. So, as I mentioned, we are now going to talk about our first major physiological process, and that is going to be mitosis. Now, mitosis is a smaller portion of a much more important physiological process known as the cell cycle. Here we have the pretty picture from your textbook that talks about the cell cycle. And basically, as you can see in the term cycle as it represents, it is a continuous process. And this continuous process basically has two main parts to it. So in our cell cycle, there are two main parts. Oh, I guess it helps if I'm using a color we can see now. Excuse me. And those two main parts are interphase, and the other part is the mitotic phase. All right, let's talk about interphase first. If we are defining interphase, basically in interphase, there are two main processes that occur. The first is the cell is doing all of its normal cell stuff. If it's a toenail cell, it's doing toenail things. If it's a liver cell, it's doing liver things. If it's an eyeball cell, it's doing eyeball things. It is doing all of its normal functions. However, on top of that, the other thing that our cells are doing during interphase is they are preparing to divide. Again, all cells come from pre-existing cells, so when a cell is getting ready to divide, that process of getting ready to divide occurs in 3 steps, as we can see here. The first step of interphase is what is known as G1. During the G1 stage, let's capitalize that. This is the longest of the stages and the most variable. For some cells, G1 may take, as you can see here, 8 hours. In others, it can take months to go through this phase. So again, it is the longest and the most variable of all of these stages. And as you can see here, basically, G1 really stands for growth, right? This is when we get the growth of the cell. Remember, if one cell is going to become 2 cells, if they're going to be functional cells, they need to have rough endoplasmic reticulum. They have to have a Golgi apparatus. They have to have mitochondria, right? So if a cell, and let's go really simple numbers, if a cell needs 2 mitochondria to survive, and it has 2, and it divides into 2 cells, and each cell only gets 1, then those new cells aren't going to have enough. So what happens is that cell first has to go from 2 to 4 mitochondria, so that when it divides, both cells get the 2 that they need. So this is when the cell is growing. We produce more organelles. We are going to start protein synthesis. Remember, as we said, we're not going to be storing those Christmas lights all year round. There are lots of proteins that are needed for cell division. So we start protein synthesis here. But there's one other really important process that starts here. We start the process of replicating the centrosome. Remember, as we mentioned, the centrosome is what is responsible for moving the chromosomes around. It's that microtubule organizing structure that is going to move the chromosomes around that are required in our cells for cellular division. We start making the centrosomes, a second centrosome, replicating it here in G0. And it's actually the end of that process that is one of the big triggers of what ends interphase and then starts the mitotic phase. All right, questions on that? All right, the second phase of interphase is what is known as the S phase, or the S stage. Let's be consistent. In this case, S stands for synthesis. We're synthesizing things, or really what's really happening here is here's where we replicate the DNA. After all, if a functional cell is going to have 46 chromosomes, then when we make 2 cells, we need to make sure both cells have 46 chromosomes, right? And it's a little more complicated than that as well. If I have a deck of cards, right? How many cards are in a deck of cards again? No gamblers out there?
52.
52, absolutely. So if I get 2 decks of cards and just shuffle them together, I now have 104 cards. If I go 52 on one side and 52 on the other side and I just do them randomly, are those going to be functional decks of cards? They have 52 cards, but are they necessarily going to allow me to play the games that I want to play? No. Not only do I have to make copies of all the cards, but I have to make sure that both sets get an ace of spades. Both sets get a 2 of clubs. Both sets get a 3 of diamonds, and so on and so forth. So not only do I have to make copies of the DNA, but I have to keep track of them to make sure that one goes to one cell and one goes to the other. So that's what's happening during the S phase. We're replicating and keeping track of the DNA. And that then brings us to the G2 stage. In the G2 stage of interphase, this is where we're going to make all the special proteins that are going to be necessary for division. There are hundreds of proteins, different proteins that we need numerous copies of. And this is where we make all the special proteins that are going to be used just for cellular division. Cellular division requires a lot of energy, a lot of resources, a lot of ATP, a lot of time. So again, it's a special process and we don't want these things hanging around all the time. So we have those special proteins that are made. And as I mentioned, at the end of G2, that is where we finish, let's say it that way. Or yeah, let's say finish. Replicating the centrosome. And once we finish replicating the centrosome, that is going to be our trigger to start mitotic phase. All right. So notice a couple quick things about this. Again, it's much more varied in the time that it is going to take. 3 big key phases to them, starting with G1, the longest, the most variable, then the S phase, and then the G2 phase. And then at the end of G2, once we've finished replicating the centrosome, then that is when division starts. And again, notice also, we've already replicated the DNA. Remember, as we talked about, if the cell's going to divide, we need to take that loose chromatin and fold it up into chromosomes. If I take a piece of paper and I fold it up 15 times into a tiny little ball, is it easy to read that paper and make a copy of it? No, once it's folded up, I'm not going to be able to read it and make a copy of it. So it makes much more sense to make that copy when it's unfolded, when it's uncoiled, when I can actually read it. And so that's why notice we actually make the copy of the DNA during interphase before division begins. All right? Questions on that? All right, that is interphase. Let's go ahead and clear the picture for that. So then we can talk briefly about mitotic phase. Again, as I've mentioned, there's actually 2 things. I don't know if I've mentioned the second thing yet. As we've mentioned, anatomists love to name everything. The second thing you need to know about anatomists is that they hate you. And again, one of the ways that they express that hate for you, as we talked about, is they come up with rules and then they break those rules all the time. Another way they do it is by having numerous names for the exact same thing, like anterior and ventral mean the exact same thing, like Golgi apparatus and Golgi complex mean the exact same thing. Well, here's another example. This next part of the cell cycle is called the mitotic phase. The mitotic phase is when the cell divides. But here's the problem. The mitotic phase involves 2 overlapping processes. Notice interphase, each phase was separate from each other. Not only were they separate from each other, but there might be a break in between those phases when they occur. Mitotic phase has 2 processes that overlap in time. And the problem with the mitotic phase is that one of those phases is mitosis, but the other one isn't. It's called the mitotic phase because this involves mitosis, but it's not mitosis by itself. Right? It's kind of a crummy way to think of this. So in some ways, instead of mitotic phase, if we think of it instead of cellular division, right? If it's the cellular division phase, then it would make much more sense than just the mitotic phase, because mitotic phase involves 2 processes. Mitosis is one of them, and the other, as you can see from the illustration, is cytokinesis. So if cellular division involves 2 processes, mitosis and cytokinesis, then one of the things we need to do is define these 2 processes. What is mitosis? What is cytokinesis? Anyone know what mitosis actually is? What actually happens in mitosis? I'm sorry. Go ahead.
The splitting of a cell?
No. See, again, and that's part. Mitotic phase is the splitting of a cell, but what actually happens just in mitosis?
A copy of the cell.
No. In mitosis, actually what happens is the splitting, not of the cell, but the splitting of the nucleus. Mitosis is when one nucleus becomes two nucleoli. If mitosis was all that happened in the mitotic phase, then at the end of the mitotic phase, what you would do is you would have one cell, and that one cell would have two nuclei inside of it. Mitosis is when the nucleus is split into two different nuclei. Cytokinesis is actually where we split or divide the cytoplasm. So cytokinesis is actually where we physically cut the cell in half. So the mitotic phase, to get two successful cells from one, requires two processes. First, we have to take one nucleus and make two nuclei. Then we divide all the cytoplasm in half and physically divide it into two cells. So actually cytokinesis is where we actually physically divide the one cell into two. And mitosis is actually where we split the nucleus. All right. Now notice also, just to add layers upon layers, notice when you look at mitosis, mitosis has 4, or depending on how you think of it, 5 stages in it. And those, as you can see, are prophase, metaphase, anaphase, and telophase. I just abbreviated here because they have it written out on the illustration next to us. And because I like my mnemonics, if you ever have trouble remembering what the 4 stages of mitosis are, just remember what it is that you have in your bathroom. In your bathroom, you have a PMAT. Right? And so that PMAT helps you to remember the 4 phases in order for mitosis. Notice also, as I mentioned, these are overlapping processes. It's not like mitosis happens first and completes and then cytokinesis occurs. Notice cytokinesis starts about halfway through anaphase. And even though it shows it here as stopping at the same time as telophase, it's actually possible that cytokinesis can actually extend beyond telophase as well. So it can go a little past telophase in its process as well. All right. We are going to talk about all of these in much more detail, but before we get to that, we have to come back here to the S phase and talk about the replicating of our DNA. Because we've talked some about our DNA, but we need to talk a little bit more. All right. However, if you notice as you look on this illustration, we have this continuous cycle, but notice also we have this funny little box out here. This funny little box is outside of the cycle. And this funny little box outside of the cycle, it is what is known as G0. G0, and again, this is not when the cell dies. This is not when the cell goes to sleep. But what G0 is a state outside of the cell cycle. Again, in a cell cycle, it is a continuous circle where it is continuously going around. You get ready to divide and you divide. You get ready to divide and you divide. But G0 is a state outside of that cell cycle. Almost all cells spend at least a little time in the G0 state. In this G0 state, the cell is not dead, it is not asleep, it is not hibernating. The cell is completing all of its normal life functions. In fact, that sounds an awful lot like interphase. So what's the difference between interphase and G0? Well, in G0, the cell is not preparing to divide. Now, let's go back. Notice I said almost all cells spend at least a little time in the G0 state. There are a few cells that as soon as they get done dividing, go right back to getting ready to divide again. What do you think we would call one of those cells that as soon as it got dividing, got started on dividing again immediately? I know you guys know the answer, you just haven't thought of it this way. What cells did we talk about? Don't really have any other jobs except dividing to make new cells?
Stem cells.
Stem cells, absolutely. So notice our stem cells spend little to no time in the G0 state because they are getting ready to divide almost immediately. As soon as they're done dividing, they go back into it. But pretty much every other cell will spend some time in this G0 state. And some cells spend more time than others. Let's talk about your liver cell, for instance. A liver cell spends about 1 year in this G0 state. So as soon as a liver cell divides, it enters that G0 state where it's doing all of the functions of your liver. Your liver cells have over 200 functions that they're responsible for. They're not hibernating, they're not sleeping, they're very, very busy. And it's chilling in that G0 state, doing all of the jobs that it needs to do, but it's not thinking about dividing. Then after about a year, it exits that G0 state and gets ready to divide again. All right? This is why if you damage your liver, you decide because you've been home for 90 days now and you're doing nothing but drinking breakfast, lunch, and dinner for vodka. Wait, the other way around. Drinking vodka for breakfast, lunch, and dinner. You're probably doing some serious damage to your liver. Now, with enough time, with years, could your liver repair itself? Yeah, but if you've done serious damage, do you have the time to wait for it to repair itself?
No.
No. So what ends up happening is you need a liver transplant. And even before, you would get a whole liver. But now they've gotten more sophisticated. What'll happen is when a person typically donates a liver, they don't donate their whole liver. They just donate a portion of the liver. So you get a portion of one person's liver and they keep a portion of theirs. 10, 15, 20 years later, eventually that part of a liver will grow into a full liver. It's not going to do it overnight, but eventually after years and years, you and the donor will both have 2 full-size livers at the end of that. But it's a slow process. Other cells, like we talked about our skin cells, right? They're so rapidly dividing, producing millions of new ones every day, they spend very little time in that G0 state. However, as we talked about earlier, there are 2 cell types that when they mature, they enter the G0 state and never leave. Meaning that they are never capable of dividing and regenerating and replacing them. And what were the 2 tissue types that we talked about? There you go, nerve tissue and?
Brain or heart?
Heart, cardiovascular, there you go, cardiac muscle. Those 2 tissue types, they enter that G0 state and they never leave it. They are not capable of dividing anymore once those tissues have matured. All right.
So if they do damage, they will most likely not repair.
Exactly, right? If you are in a severe car accident and you have a traumatic brain injury, right? Typically, well, again, the original trauma, often from the original trauma, there is some improvement that occurs from that. But like I said, if you get a spinal cord injury where you become a paraplegic, right? Is that ever going to heal on its own after that? No, you're going to basically spend the rest of your life in a wheelchair. Right? Cardiac muscle becomes damaged, that tissue is damaged forever, right? And if it upsets the function of how the heart works, then we have to put in a pacemaker to make the cells of your heart function properly so that it can be fixed, or we have to get you a new heart. You know, those two tissues aren't capable of repairing themselves. And as someone pointed out earlier, those are two of the most important tissues. All right? So we did that, excellent. We've talked about all of that. So now, like I said, before we can talk more about mitosis and cytokinesis, we need to come back to this replication of the DNA. We've been slowly adding layers of information about our DNA, and so we're going to add to it now. We know DNA is comprised of nucleotides. Those nucleotides, as we talked about, have those three components. And those are a sugar, that negative phosphate group, and a nitrogen base. As we hinted at, those sugars and the phosphate form a backbone. So basically, when you look at the DNA, what happens is we get this backbone that is made up of the phosphates and the sugars, and then coming off of that is where you have the bases. And remember, as we mentioned for DNA, those possible bases are the adenine, the guanine, cytosine, and thymine. And again, I'm just writing them as the letters here because you see them to the side there. Remember, as we said, if instead that sugar is a ribose, then we've got RNA, and it uses adenine, guanine, cytosine, and uracil. All right, so we have this phosphate sugar backbone, and we have the bases that come off of that. But as it turns out, there are DNA forms what we call a double helix. And so what that means is there are actually two sugar and phosphate backbones with bases that come off of them. And bases form pairs, what we call complementary pairs. Not just any old nucleotide can match up with any other one. Anyone know what A matches up with?
G.
C.
T. Adenine and thymine match up. What does G match up with?
C.
C.
So then C matches up with
G.
And T matches up with A. Excellent. Notice as we look at these pairs, while these nitrogen bases are all nitrogen bases, there are two different types. Some that are larger, made up of two nitrogen rings. These larger bases are called purines, and the two purines are adenine and guanine. And notice we have the smaller ones made up of a single nitrogen ring, and those are cytosine, thymine, and uracil. So notice when we have our pairs, we have one large and one small. One large and one small that always match up to form these complementary pairs. The reason this occurs, which one can match with which one, oh, where's my picture? Oh, there it is because they can form different number of hydrogen bonds. So a purine always matches up with a pyrimidine, but notice that adenine and thymine are both able to make hydrogen bonds, whereas guanine and cytosine are both able to make 3 hydrogen bonds. So what we have are these specific rules on how these are going to match up. And we can understand that when we look here at the pretty picture of that double helix, where we have that sugar and phosphate backbone. And notice you always use those complementary base pair rules. So notice if I had a strand of DNA that was comprised of A, C, G, T, C, G, T, A, something like that. If that was one strand, what would the next strand be? What would be complementary to the A?
T,
T, excellent. Then what's next?
G,
Don't make me keep saying what's next.
C, A, G, C, A,
T.
T.
T, excellent. Perfect. So the good news is when we know what's on one strand, we always know what's on the other strand. But the other important thing to remember is the thing that is holding these together are hydrogen bonds. Now, again, I know A and T have 2, C and G have 3, but I don't have enough space between them to do that. So I am just going to draw the hydrogen bonds as red lines. And as we talked about before, are hydrogen bonds really, really strong or really, really weak?
Really weak.
Really weak, exactly. And we can use that to our advantage. The exact same way that I gave you a strand of DNA, and because you knew the complementary base pair rules, you were able to give me the other strand. That is exactly what the cell does when it comes time to divide. When we need to replicate the DNA, what it does is literally exactly what we just did. Because these are hydrogen bonds, what I'm able to do, oh, gotta select, no, come back. Hold on, back to there, no. That, stop it. There, excellent. Grabs one of the strands and rips it apart from the other ones, breaking those hydrogen bonds. Let's get rid of those hydrogen bonds. Then, using our complementary base pair rules, I am able to make a new strand of DNA using those base pair rules. So again, what goes here?
T, G,
D, G,
C, A, G, C, A, T.
Excellent. And what goes here? Oh, here, I know we can't see it. Or at least I can't see it. There we go. What goes here?
C, G, A, just kidding, T, C, G, T.
Perfect. Notice where I had one strand of complementary DNA before, now I have 2. Each one uses one of the original strands, and each one uses the base pair rules to make a new one. And it literally is that simple. Of course, it's really literally not that simple. It's an incredibly complex process that uses dozens of proteins and dozens of nucleotides and dozens of enzymes and dozens of everything. But that's essentially exactly how we replicate our DNA. Now, as I mentioned, there are dozens of enzymes involved in this process. But the most important one, the one that does the important work that you and I just saw, is the enzyme DNA polymerase. Now, this also gets us into the 3', 5' rule. Again, I know you're going to get all of that in micro, so I'm not going to worry. We're not going to worry about that rule. But what I do have is a pretty picture that shows exactly what is involved in this process. Notice here it shows a couple of enzymes. This one helps to unwind the DNA, breaking the hydrogen bonds. There are other enzymes that bring the nucleotides and all those types of things. But if you notice right here and right here, these two are the important enzymes, the DNA polymerases, that are doing exactly what you and I did. If they saw a C, they added a G. If they saw an A, they added T. And so where we had one strand of DNA, we end up with 2 identical strands of DNA. And so this, again, remembers, happens during the S phase of interphase. In the S phase of interphase, we replicate the DNA, making 2 identical copies. And that is our DNA polymerase that does that. All right? Questions on that? All right, more fun with vocabulary. Again, remember, this is happening during S phase when our DNA and proteins, remember chromatin is DNA and proteins. You can't get DNA by itself, so it has to be DNA and proteins. Collectively form our chromatin. So when it's loose, chromatin is when we make the copies. When it comes time for it to divide, that chromatin is then folded up into our chromosomes. All right, no new vocabulary there, no new concepts there. You with me so far? And here we have the pretty picture that shows this. Our loose chromatin made up of DNA and proteins like those histones are folded up into a tightly packed structure that is a chromosome. But now we have additional information. We understand that when this chromosome forms, it is replicated. And we have 2 copies. You may not have thought of it in these terms, but you are aware of it because when most people draw or show chromosomes, how do they draw or show those chromosomes? When you look at a chromosome, what does it often look like?
A pair?
Well, yeah, it's true, it's a pair, but often when you look at it and they're drawn, they're shown as X's, right? You've seen that before?
Yeah.
The big X as the chromosome. That's not actually what it is. Let's do a more accurate drawing of it. So let's actually just erase my X and do a more accurate drawing. What it actually is we have one strand of all that DNA looped together and a second strand of identical DNA looped together as well. Right, so what it is really not so much of a, it isn't so much of an X as it is an H. We have 2 identical copies of DNA that are held together by a special protein. Now again, let's do some fun with vocabulary. This whole structure is 1 chromosome. Again, it always looks like an X, but it's really more of an H. This 1 chromosome is made up of 2 identical copies of the genetic material. And those 2 identical copies we call sister chromatids. So 1 chromosome is 2 sister chromatids. Now we don't want those sister chromatids getting lost. So those sister chromatids are held together by a special protein, and that special protein is called the centromere. So 1 chromosome is 2 sister chromatids held together by a centromere. Fun with vocabulary. All right? With me so far? If you're not, say now, because it's just going to get worse. All right, excellent. Notice we have our chromosome, but we know one other thing about our chromosome. We know our chromosome is going to get moved around inside the cell by those microtubules. Remember, the microtubules are going to attach to it, and we know it's going to move around the cell. It's able to do that because there is an additional protein attached to each of the sister chromatids. Each sister chromatid has a special protein on it. This is a protein that our microtubule can attach to. So I'll just attach 1 microtubule to it. And now let's go and label everything. This is a microtubule. Oops, why did that not write? Right. Or here, this special microtubule is called a spindle that is being formed there. And the special protein that it attaches to, so let's draw an arrow. I'll put it on this side. No, hold on. I'll put it down here so that we can see it. that special protein that the microtubule connects to, because it connects to it, is called a kinetochore. Kinetochore. Excellent. So fun with vocabulary. One chromosome is made up of two sister chromatids held together by a centromere, which is going to be moved around the cell by microtubules that are attached to their kinetochores. Again, I've done an amazing job of drawing this, but let's actually look with the pretty pictures within your textbook. So again, when a chromosome forms, it has already been replicated because replication takes place during S phase of interphase. So we have two sister chromatids held together by a centromere. So one chromosome is two sister chromatids held together by a centromere. And each sister chromatid has a special protein called a kinetochore that is used to move it around the cell. All right. Fun with vocabulary. Questions on that? All right. If we're comfortable with the vocabulary, then we can talk about the process. As I mentioned, and here we see a nice example of what I was saying before, what we call the mitotic phase, or really the cellular division phase, is comprised of two overlapping processes. Mitosis, which is when we divide the nucleus, and cytokinesis, which is when we actually physically divide the cell. It is when we physically divide the cytoplasm. They overlap in time, and notice cytokinesis can go beyond telophase. Mitosis has 4.5 stages to them: prophase, metaphase, anaphase, and telophase. And I'll tell you why we say in a half in just a minute. Again, the two phases overlap. Cytokinesis usually begins in late anaphase and can continue past telophase as well. Cytokinesis is when the cell actually pinches in half, right? This is like kind of like right after Thanksgiving, you put your belt on your pants, right? And you have to pull it really tight to cinch it down to make up for all of that stuffing you've been eating. And that's kind of what happens on the cell. The cell gets this ring around it. That ring happens to be made of actin. And as that actin squeezes, it wraps around like a belt, it squeezes, it pinches the cell. As it pinches the cell, it forms a structure we call a cleavage furrow. Right? Because basically what happens is we start with a circle, and that circle gets a ring of actin around it. And what ends up happening is as that ring pinches more and more and more, the cell gets more narrow in the center until eventually it pinches completely in half, and then you have two cells. Plants are very weird. Again, you are responsible for looking at onion root tips and plant mitosis because it's a great example of it. Your slides involve both animal and plant examples, so you want to make sure you look at that. But what ends up happening in plants is that instead of pinching the cell, because plants have cell walls, a little bit of a cell wall forms in the center, what is called a cell plate. And then that cell plate expands and gets bigger and bigger and bigger and bigger until finally it divides the cell in half as well. All right, so we'll look at some pretty pictures of these, and we'll talk about these as we look at it. How are we on time? We're doing good. Excellent. We're not doing good. We're way too tight on time, but we'll keep going anyway. All right, excellent. Let's talk about these processes then. Starting first in mitosis. Here is our cell in interphase. Notice we have our DNA that we've been loose in its chromatin, loose chromatin, DNA and proteins. It's already been replicated. Notice there's a nucleolus showing that we're making proteins right now. But remember, the trigger for mitosis is going to be the completion of our centrosomes. All right. Now, often when we go through these more physiological processes, what I find works best, and again, we're somewhat limited by the way we're doing this, is I like to draw it first on the board. We go through it together on the board, then we go through all the pretty pictures and all the pretty words. What I strongly encourage you to do for this first part is to just, again, if you're taking notes, which hopefully you're taking notes, put your pencils down, just watch, look, think, and listen. I just want to go through it once while you watch and observe. And then once we're done with that, we can make more sense of it by looking at the pretty pictures and looking at the pretty words and making sure that it all makes sense that way. But for the first time through, let's just do it on the screen and make sense of it. Now, again, the first thing we have to remember is what our goal is. Our goal is to divide one cell into two identical cells. So that is our goal. So towards that end, we're going to start with a cell. And for this, let's draw a cell and give this cell a nucleus. Now, stop it. New circle, perfect. I'm going to make my nucleus big so we have some room to draw it here. Now, as we know in our cells are going to have 46 chromosomes, right? And as we also know, those chromosomes are going to be comprised of two sister chromatids that are held together by a centromere, right? Which kind of gives us that X shape to them. In the interest of time and space, I'm not going to be able to draw 46 chromosomes in here, so we'll just do 2. So our goal is to go from one cell with 46 chromosomes, or in this case, one cell with 2 chromosomes, to form 2 cells. Oops, whoa. Move that there. Circle, cell 1, cell 2. And both of these are going to have a nucleus inside, and both of these nuclei are going to, and I'll just draw X's because it's simpler, 2 chromosomes. So that's our goal. One cell dividing once, producing 2 cells with the same number of chromosomes. All right? Oh, hold on. Let's go back. Whoops. Undo. Now, remember, here those chromosomes were replicated, so here we don't want them replicated, so there you go. 2 chromosomes identical to the original one. That's our goal. And as we mentioned, this is going to take 4.5 steps. So let's draw 5 circles here. 1, 2, 3. 4 and 5. So we can identify and talk about each step. Now, as we know, the first step is prophase. But as it turns out, a lot of stuff happens in prophase. So since a lot of stuff happens in prophase, one of the things they often talk about is early prophase and then late prophase. So that's often what is done in prophase. We have early prophase and we have late prophase. Now, because anatomists love to name everything and because they want to sell new books, some physiologists have actually started talking about prophase as being its own, early prophase being its own step. And of course, remember, we know the next step is going to be metaphase. And so they said, well, you know, since this late prophase, so many important things are happening, maybe we need to give this phase its own name. And so we need a big, fancy, showy name for something that happens between prophase and metaphase. And because they're not very original, they came up with prometaphase. So depending on your textbook, depending on who you speak to, you may hear mitosis as having 4 or 5 stages. And you may describe it either way. You may either go early prophase and late prophase, or you can say prophase and prometaphase. I am fine either way you want to do that. Okay, but let's talk about what happens. in prophase, there are basically 3 main processes that begin in prophase. And I'm running out of space already, so I'm going to cheat and get rid of my goal pictures because it's annoying and it's in my way. You know what the goal is. So let's get rid of all that because I need the space. And that'll allow me to move this up here and then this. I can cheat more into this space. Excellent. There are 3 main processes that begin in early prophase. Again, remember the trigger for this, and I'm going to draw them as just 2 lines, is that our centrosome has replicated. Now, when these centrosomes have replicated, the first thing that's going to happen is that our centrosomes are going to migrate to opposite sides of the cell. Right? Those centrosomes are going to start to move. Right? So this one is going to start moving this way. This one is going to start moving this way. And remember, these centrioles are the microtubule organizing structures. So the other things that's going to start to happen is they're going to start to form microtubules around them. They're going to start to form this kind of star-shaped microtubule structure around them. And these are the beginning of the formation of those microtubules, or remember what we call the spindles. So our spindle fibers are going to start to form. All right, so that's the first main process that begins in prophase. The second thing that happens in prophase is that our chromatin is going to fold up into chromosomes. So we have that nuclear envelope still where we have the nucleus inside here. And inside here, that loose chromatin is going to start to condense down into our chromosomes. And there are a couple of there's one starting to form. Here's another one starting to form. And then the third thing that happens is if we're going to move those chromosomes around in the cell, we have to break down the nuclear envelope. We have to get rid of the nuclear envelope because otherwise, if the chromosomes are still locked up inside the nucleus, then there is nowhere for them to go. So these things have to start breaking down. And so these 3 processes, and again, the key is they all begin during early prophase, but they are not completed until late prophase.
Hey, I'm in class. I'll call you back.
I'm sorry? Okay. So they start these processes in early prophase, and by the end, by late prophase, all 3 tasks are accomplished. Our centrosomes have reached the poles. So, and let's just do, we'll do left and right. Centrosomes have reached the poles. And again, I'm going to change the color of them. I know my colors aren't consistent, but that's all right. We have those spindles that are coming out from them. Our nuclear envelope is completely broken down. And our chromosomes are fully formed. So we'll put one of the chromosomes over here, because again, without the nuclear envelope, these things are going to be able to move wherever we want them, wherever they want. And this one's going to be formed here. So notice we started these processes in early prophase, and in late prophase or prometaphase, these 3 processes have been completed. But there is one other important thing that occurs in late prophase. The other thing that happens in late prophase is that those spindle fibers are going to attach to the chromosomes. Right? And they do that, remember, on the kinetochores. So while some spindles are just going to be helping to maintain the shape and structure, from each side, and these need to be yellow, a spindle is going to attach to this one, and a spindle is going to attach to that one. A spindle is going to attach to this one, and a spindle is going to attach to that one. So all of our chromosomes are attached to spindles now by the end of late prophase. Once this occurs, then we move into metaphase. The goal of metaphase is to line up all the chromosomes on the, and I'll put it in quotes here, equator of the cell. If the centrosomes are the poles, let's try to be consistent with the colors. If the centrosomes are the poles, there's your north pole, there's your south pole, then the equator is going to be the center part. And of course, it's not called the equator. We're going to use the correct anatomical term, and that correct anatomical term is the metaphase plate. So what's going to happen is our chromosomes are going to line up along our metaphase plate, right in the center. To do this, what happens is that some of our spindle fibers are going to get shorter. So when they get shorter, they pull it in one direction, and other spindles are going to get longer, so they push it in that direction. So notice this one would have to get longer, this one would have to get shorter to bring it to the middle. Whereas it's the opposite on this side. On this side, this one had to shorten to bring it to the middle, and this one had to go longer to bring it to the middle. And so by dynamically changing the shape of these microtubules, we can line up all of our chromosomes on the metaphase plate. So metaphase is about lining up our chromosomes. That brings us, and again, let's draw a line here just so we're not confused by this, to anaphase. In anaphase, once all the chromosomes are lined up on the center, on the metaphase plate, what happens is that centromere, remember the centromere is the protein that's holding them together, it inactivates. And when it inactivates, the two sister chromatids let go of each other. Now they're no longer connected. Remember, they were sister chromatids held together, but now they become what we call daughter chromosomes. Oops. So what were sister chromatids are now going to become daughter chromosomes. And those daughter chromosomes, I'm just going to DC it, abbreviate it for space, are then going to be pulled to the poles by the spindles. So what happens here, and I've got my centrioles, oops, there and there. I got my other centrioles for the centrosome right there and there. And the centromere has inactivated. I need yellow spindles. Those spindles are going to start to rapidly shorten. And as they rapidly shorten, they pull the now daughter chromosomes to the opposite sides. It is a dynamic, rapid process. And so what ends up happening when you look at these under the microscope, you can actually see that the chromosomes go from the X shapes that we had before to these very distinct V shapes as they're being pulled towards the poles of these cells. So we get these very distinct V shapes to these daughter chromosomes as they are getting pulled to the opposite sides.
Professor?
Yes.
Maybe I heard wrong, but did we talk about before like sister And now we're talking about daughter. What's the difference between them?
When they're still connected together, they're sister chromatids. But once that centromere inactivates and they are no longer connected, they are now considered daughter chromosomes.
Oh, okay. So once they're pulled apart, they become daughter.
Correct.
Okay.
Technically, once the centromere inactivates. When the centromere inactivates, they are no longer attached. When they're no longer attached to each other. But as soon as they're no longer attached, they get pulled to the opposite side. So yeah, when you see them separated like this with the V shape, they are now daughter chromosomes. All right. That brings us to the last stage. The last stage is, of course, telophase. With telophase, this is basically, whereas in prophase, we broke down the nuclear envelope, we folded up the chromosomes. Here in telophase, we are going to reform, and not one, but we are going to reform 2 nuclei, right? So in some ways, it's kind of like prophase in reverse, right? One of the things we need to do is we need to reform, again, 2 nuclear envelopes. We need to unwind the chromosome back into chromatid. Back into chromatin, right? And, oh, no, don't want green, hold on. Let's go with red. So now we have our nuclear envelopes reforming. Our brown DNA is now loose. Oh, wait, hold on. No, I didn't want that one. That one. Now it's this loose squiggly chromatin again. Okay, contained in the nucleus. Pretend that's contained in the nucleus. And remember, now that it's loose, this is also when we can start getting those enzymes around it to start making proteins again. So this can actually be when nucleoli will start to reform, which tells us that we are making proteins. And at this point, we have accomplished our goal of mitosis. Remember, mitosis was taking one nucleus and dividing it into 2 nucleoli, or into 2 nuclei. Now, remember what goes along with this, related, overlapping, is cytokinesis. Cytokinesis, remember, is when we physically divide the cell in half. This starts in late anaphase, and if you notice, you can kind of see why that would make sense for this to start to occur in late anaphase, because in late anaphase, right, there's nothing in the center of the cell anymore. So what we have is we have an actin ring, and that actin ring is going to start squeezing, start pinching that cell. So what's going to start to happen is that our plasma membrane is going to start to indent. And as we get that indentation in it, remember, it's what we call that cleavage furrow. That cleavage forms until eventually that actin ring squeezes and tightens and pulls the entire cell in 2, at which point we now have 2 completely separate cells. And one cell has become 2 cells. All right. Questions on this? All right.
Yeah, I have a quick question. You said before about the nucleoli. That means just 1, right?
Nucleolus is singular, nucleoli is plural.
Okay.
All righty. Let's go back to the pretty pictures from your textbook and do the whole thing again. Perfect. All right. Again, here we have early prophase, or really here what we have, let's start first. What we have here is interphase. Chromatin in the nucleus, 1 nucleus, making all the proteins. But again, here is our trigger. Our trigger is when that second centrosome forms. When we get that second centrosome, that triggers the beginning of the mitotic phase, the beginning of cellular division. It starts here with early prophase. And again, notice here in early prophase, as we talked about, I got to bring my annotation thing back. In early prophase, there are three main things that happen. And again, in early prophase, these three things start. The first is the migration of the centrioles, right? And formation of the spindles. So notice we see that happening. They're pushing away from each other. They're making those microtubules, those spindles that are forming. That is the first thing. That is thing one that is occurring. The second thing that is starting is we start to fold up the chromatin into chromosomes. So notice it's starting to fold up. And again, oh, I don't know why that stopped writing. Where did that go? Oh, well, let's do this. So 2, folding of the chromatin into chromosomes. And again, notice if we start folding it up, you can't make proteins anymore. So as was just mentioned, our nucleoli go away because we can't make proteins anymore if we're folding up our chromatin into chromosomes. And of course, the third thing that needs to happen is we need to break down the nuclear envelope. If we're going to move the chromosomes around in the cell, we can't keep them locked up in the nucleus. So in early prophase, these three processes begin. All right? So our nucleoli disappear, centrosomes start to migrate, nuclear envelope starts to break down. All right? Questions on that? All right. Those processes start in early prophase and they end in late prophase, or if you want, what we call prometaphase. Notice our nuclear envelope is now completely broken down. Our chromosomes are completely formed, and our centrosomes have reached the 2 poles of the cell. So we have accomplished our goals. However, remember, a fourth thing occurs in late prophase, and that's what we're seeing right here. Those spindles are going to attach to the kinetochores of the chromosomes so that they can be moved around the cell. So our spindle fiber is attached to the kinetochores so that these chromosomes can be moved around, one from each side. Notice here, when we look at a real cell that has been colorized, we can see the spindles really nicely. The nucleus is gone, right? Compare that to what we were looking at before, if you weren't looking at it before. Here you can see, well, let's actually go all the way back. Oops. Notice here you see the nucleus, and it's just uniform cloud of DNA. This is interphase, when it's loose chromatin. Here in early prophase, notice you can start to see the chromosomes that are forming, but they're still contained within the nuclear envelope. The fact that the nuclear envelope is still here, but you can start to see chromosomes, tells you that you are in early prophase. Notice in late prophase, the nuclear envelope is gone. So now the chromosomes are spread around inside of the cell. All right? And that is our late prophase. Now, once those spindles connect to the kinetochores, then our goal is to line everything up along the midline. Line everything up on that metaphase plate. And that is metaphase. our microtubules either grow out or break down dynamically, shifting and moving so that all the chromosomes line up on the metaphase plate. Basically the equator between the two poles. Now, this happens to be a plant cell. Plant cells have a ton of DNA. So notice while you can see things are lining up on the metaphase plate, you can see some of the strands kind of sticking out other directions as well. When you look at an animal cell, the animal cells, the chromosomes are much, much smaller. So they line up perfectly on the metaphase plate. Super easy to tell. This is, though, is certainly much more condensed than that is. So hopefully you can tell the difference between prometaphase and metaphase pretty easily. And notice it's a singular structure. There's no divide in between. Notice when we get to anaphase, we can clearly see the divide in between. Because in anaphase, remember those centromeres inactivate. Our sister chromatids are now daughter chromosomes, and they're getting pulled to the opposite sides of the cell, forming that distinct V shape. Once they get there, then we have telophase. In telophase, we reform the nuclear envelopes, 2 now, not 1. We unwind the DNA back into chromatin from chromosomes, right? And we can actually start to see nucleoli form as we're able to start to make new proteins. Notice also here, they give us a nice representation of our cleavage furrow that is forming from that actin ring, representing cytokinesis. Remember, cytokinesis is going to start in late anaphase and continue into telophase until the one cell is divided into 2. And so our end result of this is to have 2 cells identical to the original one. All righty. Questions on that? All right, to again help you beat this dead horse and make sure this makes sense of this, make sure you do your Labster mitosis. It is due tomorrow. So if you haven't done the mitosis Labster, hopefully this is enough information, and hopefully that will help you to make sense of this process as well. All right, any questions? All right, excellent. Thankfully, that is all we have to talk about today. Looking at it tomorrow, we probably won't have to take as long to get through the material. It'll depend on how tricky the membrane transport stuff is. But I know today was a long day. I appreciate your patience on this. But like I said, this is the problem with packing it together. It's exhausting. So take a good 15, 20-minute break and then dive into the homework that you have due tomorrow. And I will see you tomorrow. All right, any questions? All right, I think everybody's properly exhausted. So excellent. All right, you guys have a very nice day. Stay safe, stay healthy, and I will see you tomorrow.
All right, sounds good. Thank you. Bye-bye.
Thank you.